Okay, so this is the third and final day of the winter 2023 collaboratory UCLA QC Bio workshop 29, demultiplexing QC. And we've got a shorter list. You know, the day 3 is not going to be as long as the first two. Okay, so we're going to flip back and forth between some windows. So I guess I'll leave this not in full screen, and we'll go from here. You should have another email today with the slides for today. Okay, so yeah, we kind of left off. We were talking about this paired end example. So the way you start with a paired end run, not paired end run, the dual index run. The run could be single main ends or paired main ends, doesn't matter. The indexing is dual. And the way you handle a dual index run is simply by first treating it as two independent single index runs. You treat it as a single index from I1 and a single index from I2. And you just try to figure out which is the set of distinct indices that's going to be your targets for each of those, without worrying yet about how many libraries are associated with each one of those. And if you're in a unique dual, then either the first or second index will be enough to distinguish the library. But people run a lot of non-unique duals where some of those indices are shared among multiple libraries, but it doesn't really impact the analysis as in terms of the I1 and I2 separately. At that point, you're worried more about the sequences and not their interpretation of the library. So yeah, so the first step is to just treat it like we did the single index example. You do that for I1 and I2 separately. Then you start looking at the pairs. So here is the top of the pairs histogram, and the A's were we had a list of however many it was. Well, A, B, C, D, E, F, G, H, I, J, K, L, M is 13. So we had 13 targets for I1, and it's 1 to 7 targets for I2, or eight targets for I2, I guess. Yes, 7. So yeah, so that's what it is. So the grid is that size, by 8 here. So, and you know, one of those pairs, the dark first index, and this index right here, that's for phi x. And then we also actually see some poly Gs in I2, which isn't expected. That's the eighth target. So the 6 are from the user. 7th was a phi x. 8th was this extra poly G dark in I2. And the users ones, I think, end up being all of these for the first index, and then the bottom one is the phi x one, which is dark there. So that's where the list, the sort of the targets came from for them independently. And the top of this histogram is completely the top of the Paris histogram is completely explainable in there. You're not seeing any new sequences magically showing up that aren't found by that separate analysis, which is typically the case, because if there kind of was one high up in the pairs, it should have shown up in the I1 or I2 analysis as a high one, right? You know, if it's high in the 2D world, when you project it down to the marginals on the 1D world, it's also got to be high. So you should discover it in the I1 or I2 independent analysis. And so some of these end up being just low mismatches or even an indel away from the targets, but no problem. You know, if I5 is a real user guy, and this is an I5 that's 0 mismatches to the I and 1 mismatch to the 5, no problem. We interpret him as an I5, no problem. What was more interesting is that sort of we were getting more pairs than we were expecting. Like the user's only expecting, 12, and then you know, maybe phi x would be 13, but kind of that's this list at the top, and then we just keep going. We just keep getting new pairs that we haven't kind of expected before, right? And we're still in the, you know, the 400,000s or something. Yeah, I mean, they're not as big as these. These are 70 millions and 20 millions and whatever. So these are down, but of course there's a lot more white squares than the user squares. And so if you spread out even an equal amount of the expected ones to all the white squares, that would already make you lower. And we talked about how index hopping, you know, if it's like 1%, that could still be of some concern or whatever, right? Or 10, you know, if it's, you know, whatever. So these numbers may look low here, but yeah, kind of remember they have to kind of scale them up as sort of like the amount of white space that's there because they're being spread around there. So these count more, you know, the total mass of all these other guys is going to add up to some millions. So, so anyway, so then the idea was to do in R, the same kind of thing that we did for the first guy. In particular, we want to know, what mismatch tolerance should we allow? Are there any samples, are there any individual indices that are going to need some extra targets, or, what's going on there? The same kind of thing we did there. And then add to that analysis in terms of looking at the pairs. So we actually want to see this full grid loaded up in terms of how many reads we really get landing in each of these squares once you allow the Demux tolerance that you're going to allow for the first index and second index. So we don't really see that yet, because this histogram, the 2D histogram, the paired histogram goes on for quite a while. I think it's about 1/3 of a million entries in this histogram. And that's because there's just a lot more combinations you can have. It was like, our first example was like, what, 30,000 sequences or something, distinct sequences in the first one. And well, now you can imagine it's kind of like it's squares, because it's like you kind of can put any of those with, you get kind of a big list for I1 and a big list for I2, and then you start getting all combos. So yeah, so this histogram is quite long, and we don't, you sort of really want to add up the guys with the Demux tolerance into this grid before you really make your conclusions. And so that's hard to do. It's just looking at the histogram. You want to do that in R and do it for real. So let's get our R back loaded up the way we had our R environment going with some functions loaded and libraries loaded and stuff. So we got to get back to where we were. So there was this little helper function we had loaded, and then there were some libraries we were using. So we'll get those in the environment. And then to make that little network thing that I was doing yesterday a little more convenient, we'll make this little helper function. And as an example of its use here we can just do a little example here. And you can see that, this is just read delim, which is one of the many functions in R that reads from files. This is normally where you would put the file name, and then you put your options. Like here, I'm just saying that there is no header line in this guy. The data starts right on line 1. And instead of putting a file name, I can just call my little Hoff helper and actually give it a path name that's on Hoffman. And then this thing does the little SSH pipe trick so that R will launch the SSH command. The SSH command will start a cat command on Hoffman with the path that you passed in. Out will come the contents of that file, and that'll come back through the pipe, and that's what the read delim will read from. And yeah, so if you don't have that all set up on your own situation or something, then all you got to do is wherever I've got these little Hoff parentheses, you just replace that by the path on your local machine where you downloaded the file to, and you've got to go manually pull the file and get it on your local machine by whatever mechanism you want. I assume since you all use Hoffman, I assume you all have mechanisms for transfer of files up and down from Hoffman. Okay, so the first part of this is to just copy what we had done before, but since we have to repeat it, like we have to do the I1 analysis and the I2 analysis, rather than just sort of doing the code twice and making the changes, whatever, it's a little more useful to just sort of generalize the code to be a function, and then you pass in the arguments, and then we can just call the function twice. So the first thing we'll do here is to load in our histograms. So this is a little function now that we can just sort of basically pass in the path to the histogram, and then the minimal amount of information that varies. So false here means that it's a single index histogram. True means it's a dual index histogram. And this is the labels, when we add the row labels to make the names, what prefix do you want to put on them? So we'll make sure that the I1 histogram has I1 names in it, and the I2 histogram has I2's names in it. That'll just help us when we look at tables and stuff later, because everything will have either an I1 or an I2 in front. It'll be very obvious which axes are index 1, which axes are index 2, and so on. Always a good idea to have all of your data labeled and so on, so that it just becomes very obvious in R what's what. You don't have to think, oh, rows or columns or what and something. If everything's labeled, it's just hard to go wrong. So here we're loading up our histograms. Doesn't take very long. Okay, so now we've got, that's a table basically that we did on the first example, the single index example from day 2. That's like that, but now we also have one that's laid out basically the same. That's for the second index histogram. Here's the I1, I2 name prefixes I was just talking about. But then we also now have the paired histogram. So then we have this third one, which is now got an extra column because it's got 2 mers and then the tally for that combination. Okay. And the next function we need is we need the list of targets that we chose. from the independent I1 and I2 analysis. And I've got those in 2 files on Hoffman. Those are very short, right? So there we go. We just loaded those in, and those are just the list of target sequences that we're going to demux with, so we have variables for those. Then we need to actually do the demux analysis itself, which is the thing we talked about last time. For each thing in the histogram, you go and look at the distance of its sequence to all of our targets. What's the smallest distance? Which guys are hitting the smallest distance? If there's more than one of those, then it's demux fail. Otherwise, there's a unique one at the smallest distance, and that's the pile you go to. And we just add 2 more columns to our histograms that keep track of what that minimum distance was and what demux pile it goes into. So this function demux does all that. And then we just call it twice, once with our I1 histogram with I1 targets and I2 histogram and I2 targets. So there we go. That'll take a minute or two. It's actually, this is really doing a real demux. We're not generating like the full FASTQ output files, but in terms of deciding what every single read in the FASTQ files would go to, this is that decision. The reason why we're doing it so fast in R is because we've collapsed everything by the histogram, and the histogram has entries with like 50, we have single sequences that are seen 50 million times, but our little loop inside R here only has to do that one once, right? But essentially that just figured out where 50 million guys go. So that's one of the main reasons why this is so much faster than like going through BCL2FASTQ or something, because that guy has to basically treat each one of those separately, because it's got to pick up the actual main read sequences and write the file, a lot of IO and stuff. And here, though, just in terms of deciding where they go and getting statistics about which pile, what's in, and so on, counting and so on, doing everything in terms of the histogram is so much faster, because you don't do the same thing 50 million times over when the answer is always the same. All right, so the first index analysis is done, and we're working on the second index analysis. Those text progress bars are pretty useful if you write any R code. They're very simple to use. This is the function that creates a new one, and you just sort of tell it in your loop what's going to be the bottom of your loop and what's going to be the top of your loop, and then there's some different visual styles they have which you can choose from. And then inside your loop, you just update the progress bar with this function, and you just tell it where your loop variable is, and then that takes care of converting that to percents or whatever, updating the text bar or whatever. And then when you're done, you should tell it that the progress bar system that you're done with this progress bar, and it will disappear. So it's very easy to make those, and it's a lot better than just staring at a screen waiting for something to happen and not knowing if it's like 0.001% done or if it's like 30% done, and you just got to wait a few more seconds. Okay, so we've got that, and so now we have those extra columns with our independent I1 and independent I2 histogram. Our dual one doesn't have any extra columns yet. That's our next job, basically, is to merge that information into the dual world. But we now have the information figured out on the independent ends world. But to do that, bringing it into the dual world, we're going to want to, like I was kind of briefly mentioned yesterday, you could make like the dual analysis like a 3D analysis, where one axis was the I1 target, one axis was the I2 target, and the third axis was the number of mismatches allowed, and deal with a cubical array. Those are sort of inconvenient to work with, inconvenient to visualize, and not really necessary, because we're going to have to make a decision for how much tolerance we're going to allow on the first index and the second index anyway. So why don't we figure that out now? And then when we come to make the dual world, we won't let the mismatches vary all over the place. We already know what our tolerances are. And so that'll get rid of sort of the third dimension. We'll be back down to just a 2D table, which is a lot easier to deal with. So we are now basically going to do the rest of what we did in the independent, what we did in the first example. which is we're now going to make that matrix, which is target by mismatches, right? That summary. And we'll have two of those matrices, one for the I1, one for the I2. So this summarized Demux guy is the one that does the little dplyr pipeline to make that transformation. And so we package that up in a function so we can call that twice. So we just summarize. And again, we just gets the augmented histogram and the targets, and then gives us the summary matrix out. So there we've got those. So now we have HI1 Demux and HI2 Demux, which are those two summaries. And if you just wanted a way to kind of see what the numerically the scale of those values is quickly, we could make little heat maps if you want. So, you know, the rows are the targets. The last row is for Demux fail. The columns is mismatch distance increases. So the first column is for exact hits. The next column is for exactly one mismatch away, 2, 3, 4, 5, 6, and so on. I've got the color scale and log scale here. And so, you know, this is what it looks like. Here, they're all pretty equally represented. Maybe that guy's a little lower. That's phi x, of course. And then in this one, the two that are kind of lower are the phi x is I2, and then that poly G extra one is down there too. That was these two extra rows. The bottom rows for Demux fail. Of course, you're not going to have any Demux fail with the low mismatches, right? But once the number of mismatches go up, you're going to have the Demux fail. Okay, so so let's do the plots that we did in the first time around. So we need to know the number to make things percents instead of just raw counts. We'll count the number of PF wells in the whole thing by just adding up. Doesn't matter which histogram you added up; they all are counting the same total number of reads. Here I just added up the I1 matrix. So you know this lane has 532,538,236 PF1 wells, and then we will make the plots that we did before, but now all the plots come in pairs because you do it once for I1, once for I2. So here is our little set of plots. We used to have 2 plots, now we have 4. So these first two are as you increase the Demux tolerance, and now we're talking about less than or equal to tolerance. That's what the cumulative sums did here. So like as you increase the Demux tolerance, you know, less than or equal to 0, exacts only, less than or equal to 1, the usual situation, less than or equal to 2, 3, 4, 5, 6. So on, this is the fraction, this is the percent of PF1 wells that go into a Demux pile. So in other words, not Demux fail. And so we're kind of looking here for kind of plateau, but not going too far to the right, because the further you go to the right, the more likely you're gonna have missed Demux. Now, even the exact matches only here isn't that bad. I mean, it's like 97.5% or something, but maybe, you know, this is like more than a percent gain by going to the one mismatch, and one mismatch, we're not really, you know, that's not a level you really get too excited about. You're routinely allowing mismatch. So that's probably, in terms of this view, one mismatch, not essential, but maybe okay. The gain to going to 2 isn't that much though. And it's a diminishing returns after that. Let's look at the per sample view, right? So now these are the individual, these aren't samples anymore because these are targets. Actually in this lane, since I1 is unique for the libraries, actually individual samples do correspond to this I1 plot, but in the I2 plot, these targets represent pairs of samples just due to the user's design. But you know, we don't really care in terms of what samples these correspond to, but it's just what targets they correspond to. So this is the per target view, the percent of less than or equal to 1 mismatch Demux that requires the 1 mismatch. And these are all pretty low, you know, like 1%, but maybe this one's 3.5, so there's a modest gain there from allowing 1 mismatch for target 13. It's a little more clear in the second index that maybe this one is what, it's like almost 10% that he's calling from. Anyway, probably from this, we would just say I1's going to allow 1 mismatch, I2 is going to allow 1 mismatch. But other than that, we're looking pretty good. There aren't really more I1 or I2 targets to really worry about because, you know, we're already hitting like more than 98.5% of the PF1s are going to a pile right now. And the 1 mismatch tolerance is looking okay. So that's what we'll decide in this one. I mean, usually that's what the conclusion is. It's the more rare cases when it's not the decision to do 1 mismatch tolerance. But anyway, so that will be our decision. We will do I1 with 1 mismatch tolerance, I2 with 1 mismatch tolerance. So now it comes time to work on bringing that the Demux analysis for I1 and I2 to the pairs world. So we're going to first need to kind of merge our information from those independent Demux analysis into our pairs histogram, because we couldn't summarize this pairs histogram yet until we had the extra information in there yet. So our first job is to add more columns to this pairs histogram that bring in the information from the I1 analysis and the I2 analysis. And so that dplyr can do that. And it's basically doing what in the database world or SQL world you would call a join. In particular, these are left joins because anyway, so the idea is that you have two tables which share, for example, a column that has the same kind of information in it. And you want to make a new table, a third table, which for each row of the third table, it's going to collect the information from the input tables where they have the same value in that shared column, right? So that's called a join. And there's various ways it's done depending on what you do with missing values and so on. But we have a pretty straightforward case here. So our pairs, the first columns that are going to come out are exactly the pair histogram columns themselves. So these, what you see in red here is what we're starting with as the pairs histogram. But we do have this column, which is the sequence for I1 and the sequence for I2. And then remember, we have our augmented I1 histogram and our augmented I2 histogram from the Demux analysis. And so this MER I1 can be looked up in the MER column of the augmented I1 histogram. And then we can get the three pieces of information from that guy and bring it to this row of the pairs histogram. And similarly, we can take the MER I2 and look that up in the I2 table, and then bring in the three values from the I2 table into this. Okay, so that's what all these that's what all this stuff is doing. So this first one is left joining. We're starting with the paired in histogram table, and then we're left joining in some new columns, these three new columns in orange, based on looking up the MER I1 column in the I1 table. So this is the table we're merging in, and these are the columns that are that the merge is operating upon, MER I1 on the left, MER on the right. And then the suffix actually just makes sure that we because you see this thing is called like tally, whatever, dis-mmn, dis-mmn at. Well, if we merge that in from I1 table and I2 table, we're going to have conflicts of columns that have the same name. So we suffix on the word I1 onto these names so that they become distinct. That's what this suffix business does. It just renames the column header a little bit. And then anyway, so then the second left join here is to merge in the yellow stuff. And so anyway, so that's it's very simple, really. It's just, you got to read the dplyr documentation to really understand the syntax of the joins and stuff. But logically, this is a pretty simple operation. So there we go. So we've now got into the almost 400,000 rows of the pair histogram. We've merged in for each row the information, corresponding information from the I1 and I2 analysis. Okay, now we're ready to do the summary and essentially make remember we have the plots like this from end of yesterday. We're basically ready to make the matrix, the values that get plotted in those things, which we want the rows to be the I1 targets, the columns to be the I2 targets. And at the intersection is the number of wells in the lane whose I1 index read is less than or equal to one mismatch to that row target, and whose I2 is less than or equal to one mismatch to that column target. So that's what we want in this matrix. And so we start now with our augmented pairs table. We've now got this augmented pairs histogram. And then we're going to do a bunch of stuff. The first two lines are like I talked about last time, where we don't have any holes in this thing. Is not a big deal. It's a minor point. This actually just creates a copy of columns, which just renames. We kind of have another copy of some columns with some easier to interpret names. Not a big deal either. The real magic here is, again, in this sort of group by and summarize, right? So this is the real meat of this thing. The group by, remember kind of from last time, is basically we need to group all the rows in the pairs histogram that are going to end up in the same square of the final matrix. And how do we find the rows that go into one square in the final matrix? The answer is that okay, actually, no, the thing I told you about these two lines not being that important is the wrong thing. These are the two lines that aren't really this important. These first two lines actually are important. The filter goes to a subset of rows. The two filters we impose are that the Demux tolerance is less than or equal to 1 for the I1, and the Demux tolerance for the I2 is less than or equal to 1, and it's not Demux fail. So that's basically what this first filter does, is say the I1 has to be no more than 1 mismatch to it, the targets, and it's not in the Demux fail pile. And then this one is I2 has to be no more than 1 tolerance, and the I2 can't go into the Demux fail pile in the independent analysis. Okay, then we have sort of the stuff that's less important. Now we're ready to do make the matrix. The group by pulls out which Demux pile did I1 go into and which Demux pile did I2 go into, and we're grouping by both of those variables. And that's basically which row and column are you trying to go into, right? So we now have grouped the augmented paired histogram by where you're going to end up in the final matrix. Then we need to summarize, because that's what's going to now give us the single number that goes in that square. Whereas at the moment, we have a whole set of rows that are trying to go into that place. And the group by is to take the sum of the tallies of all the guys that are trying to go there. And then we call that a new column called tally less than or equal to 1 mismatch, less than or equal to 1 mismatch, right? Because we did 1 mismatch tolerance for the I1, 1 mismatch tolerance for the I2. Then we can ungroup. And the arrange just puts them in sorted order, not so important. The next big thing that happens is the pivot wider. And that's the thing that goes from sort of the linear representation of a multidimensional array to the actual multidimensional array. So at this point in the pipeline, we basically have 3 columns that are important. We've got the I1 Demux pile that you're in, the I2 Demux pile you're in, and then the next column is the tally. We just have a bunch of it's a 3-column thing. And then we don't want them arranged like that. We want the first column to be the row index, the second column to be the column index, and then the number to go in the square, and just make a literal 2D matrix out of that. And that's what the pivot wider does. As a side effect of all this, we end up with the I1 index column as an extra column. We get rid of it, and so then we have what we want. So I can kind of show you some of the intermediate stages. So so you see what we're starting with, right? What we're starting with is this, the augmented pair histogram. But if after the 2 filters, right? So, I have to go a little deeper, I guess. So if we show like 100 lines or something, right? So if I show 100 lines, then you can see that we've got some like that start to have Demux tolerance 2 and 4 and stuff and whatever. We don't want those. So when we get to the 2 filters are done, you'll see that those are gone. And now all the Demux tolerances have to be less than or equal to 1. And any of the Demux failed pile numbers, which, whatever they are gone too. So that's the effect of the first 2 filters. And then once we finish the group by, summarize, and ungroup, then this is the 3-column thing I was talking about. We basically got row, the row index, column index, and the tally. Then the pivot wider is going to unwind that into the actual matrix. But you can see at the moment, we kind of got this extra CI1 column, and the row and column labels are not kind of very nice. And so that gets rid of the extra column. And then this drops down from a tibble to a plain matrix in R, and then we put the rows and column names back on. And so when we're done, we get this matrix, which is what we want. So we've got the I1 targets, the I2 targets, and then the cells are under less than or equal to 1 mismatch, less than or equal to 1 mismatch tolerance. How many reads would land there? So that's really what we came here for, the pairs analysis. And that's what we want to visualize with heat maps. And this p heat map from the p heat map library is a very nice function in bioinformatics. It makes pretty nice heat maps. I mean, there's a lot of options to control it, but even the defaults are pretty nice a lot of the time. And it can even do clustering on the rows and columns. All you got to do is flip a variable from true to false. It's pretty nice. So here is a linear. What is this? This is log scale colors. But you can see that like it colors the, it picks kind of a nice color bar. It actually, if you zoom open far enough, then you can actually, every square actually has the count like in the square. And then it's even got the row and column labels are labeled from the source matrix too. So that's nice. And if you turn on these cluster rows and cluster columns, I know this is a delivery guy calling me and I just cannot talk to him about it. Here you see it's clustered them in by rows and columns. It's done hierarchical clustering and then commuted the rows and columns and so on. You see that the labels are in a different order now. So it's not that exciting because of the current example, but it's at least it's paired up the guys that share an I2. When I showed you some of the fancier things that happen with index hopping like these and so on, how did I get these? These are clustered, right? So these, normally you do one of these and they're just going to be scattered dots all over the place. You may not even realize that there's block structure in there, but the hyper clustering will pull that out pretty easily. Anyway, and then the usual thing with log scale is that what do you do with zeros? I don't think we even have any zeros in here. Every square it got at least something, but an easy thing to do is just like add 1 or something, right? And then you're taking log of 1, which is a 0. And since most of the counts are in the millions and stuff, thousands, whatever, adding 1 doesn't change much anyway. That's just a typical visualization visualization thing you would do with you want to do log scale with data that might contain a 0. Then, you add a little pseudo count to it to bump the 0 up. If you go to the user sample sheet and figure out what its expected pairs were, you get these brown dots. He forgot phi x, of course, and that's this orange one that goes here. And in terms of the squares that got hit the most, it is exactly the expected pattern, which is great, right? So, bang, 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 3 here, 3 there, these 2 here, 2 here, and then those 2, and then phi x is where it's supposed to be. But then the point is that we've got all these other squares being filled in at, whatever it is, it's like some of these are hundreds of thousands, that's almost 200,000 and so on. And there's more of these squares than the other squares. So, why don't we start doing stuff like maybe we'll add up all of that stuff, find out how big it is and so on. Like let's start, we've got it all in R here, so it's not hard. So we need to know which squares are kind of expected and which squares aren't. And that just depends on the design. So at some point we have to tell R what the design, what are the expected squares here? And here I've done it by just sort of saying, 10th row, 6th column, 11th row, 4th column, these are all the guys that are expected. I added phi x here, that's 13th row, 7th column. And anyway, so we just make a little data frame that's just sort of like a little metadata data frame that just contains the information about which squares have what interpretation. So this is sample 1 to 12 phi x. This is the I1 target number, I2 target number, and just like a group label. These are all from project 1, and then this last one is phi x. Okay, so then what we can do is, first thing what we'll do is we'll change that into a 2D matrix that's an indicator matrix for where the user libraries are. So this little for loop just expands into, uses this information and just makes a new 2D matrix, which is in the same way that I've currently got the clustered one shown. Let me go back to the unclustered one. Right, so that's the clustered one. This is the unclustered one. Okay. So this is the matrix in the order that we keep our targets in. Right? So this is our at the beginning of this process, we decided what order the I1 targets were in, what order the I2 targets were in, and then this respects that order. So this matrix that we just made of zeros and ones is in the exact same row and column. It's in a 1-to-1 correspondence with this thing. And it's a 0 if it's a unexpected pair, and if it's a 1 if it's a user pair. And I think did I take phi x out of this one? Yes, I did, I think. So this is ones where there are user expected pairs. Okay, good. And so we could find out how many what's the total user pairs let's just look at user sequences. Let's not worry about the extra dark in I2. Let's not worry about things that have either phi-x I1 or phi-x I2, and just concentrate on the sub part of the matrix that's user user I1, user I2. So that's that's kind of what's in heavy black here, right? So this is the user section of the matrix. Let's add up let's add up all the stuff in there. We'll add up the red stuff in there, and then we'll subtract, and that'll tell us what the yellow stuff is in there, right? And then we can look at percents and stuff. So let's add up the red, the expected stuff. How can we easily do that? Well, it's pretty easy because we have this indicator 0, 1 matrix. And in R, if you multiply two matrices that are the same shape, it just entry-wise multiplies. So the zeros will kill, basically the counts we don't want will go to 0 because they get multiplied by a 0. The counts we do want will get multiplied by 1 and they won't change. And then we can just add up everything. And then that gives us the sum of basically the stuff that we want. So that's the plan here. We multiply by the 0, 1 indicator matrix and then add everything up. And so we know that the total mass of these expected pairs is 505 million, blah, blah, blah. What's the total stuff in the user part of the matrix? The user part of the matrix, which is outlined in black here, that is the first 12 rows and the first 6 columns. So we could take the full thing, pull out rows 1 to 12, columns 1 to 6, add that up, and that's what's in the black box. Okay, so that's everything in the user section. Okay, so that is now 511 million something. And now we can subtract, and that tells us what's unexpected in the user box. So that's the difference, and that is almost 6 million. And we could phrase that as a percent now, right? So we could do 100 times unexpected over stuff in black box, and that's the percent of user that is unexpected. And that ends up being, in this case, a little over 1%. So whether you think that's horrible or not depends on what you're doing, but basically that means that sort of like 1% of the samples are moving around, right? Now, luckily here, this guy's design is fairly low density, right? So all those things landing in the unexpected squares are not landing in the Demux piles for the expected pairs. So they're not causing misdemux. We have all this empty space, which is both sort of how we're seeing that this problem exists, but it's also simultaneously mitigating that problem because we're going to be able to, you got the wrong I1, I2 pairing. You're not going to go into my I1, I2 expected pairing Demux pile. So that's good. But you can imagine that if more squares were filled, then you're going to have hops that are landing on, from a filled square to a filled square, and you're not going to know, right? So you're not going to either have visibility into that process, and you're not going to have the defense against it happening, right? So now presumably here, you are getting cases where the hopping is causing some misdemux because he's shared some I2 indices, right? So in this second column, there's 2 guys that share this I2. So there's a chance that you could hop from this guy to this guy. But you can see that the hopping that we can observe seems to be pretty uniformly distributed here, right? So this yellow background is all roughly the same yellow. It's roughly the same rate. Presumably, it's happening the same thing from red to red at about the same rate as the places we can observe it. And so this red guy probably is leaking some fraction into this red guy and this guy going from here to here. But the fact that there's all these other empty squares in here means that it's not like every time this guy hops, it goes to here, right? It's like, it's going to be like a fraction. So if the overall hopping rate is like a little over 1%, and how many squares are here? It's like, what is it, 12? That's 12. So there's 10. There's 11 places. When this hops, this can go 11 places, of which only one of them is bad, because then it will land in another one we're expecting and cause misdemux. So we get almost another order of magnitude reduction here in terms of the misdemux, right? So what's going to go from this guy to this guy or this guy to this guy isn't something like 1%, it's something like 0.1%, because the other 0.9% is going into these yellow boxes, right? So that's good. But you can imagine that if this was more filled up or something, then the misdemux rate, you're going to fill more of that because you don't have all this ballast kind of like decoys to kind of soak it up. And you can get more than 1% as a rate. But anyway, so I'd say generically, like almost all lanes, when it's observable, it's like usually 0.1%, something like that. So that's presumably happening that's just like the routine level that's probably happening always, including in single index lanes, and whatever. In single indexing, of course, any hopping that happens, you can never observe it, and it always lands in another guy, right? You have no chance because you have no empty squares. It's kind of like a one row out of this matrix. You've got like the I2 is not varying essentially in a single index lane. It's the empty string. And it's the matrix, if you were to visualize it as the matrix, would just be a single row. So single hop carries you anywhere in that row. So you don't have any defense there. So yeah, okay. So anyway, so that's the situation. This tells you now how you can investigate your paired, your dual index lanes for index hopping, right? And sort of even kind of start to get a handle on the rate. And you can also kind of predict which samples are more likely to be confounded with which other samples, right? So for these two red guys in this column, it's much, what you're going to see if you had a way in the data to see an effect of it would be these two guys would be leaking into each other, but probably not any other thing, right? All the leakage is going to be between these sort of like 6 pairs, but between the 6 pairs and other pairs, there won't be so much leakage, right? That's going to be another order of magnitudes down below. So yeah, so we already kind of talked about. We actually, I kind of did it back in a different order yesterday. I moved this kind of the slides that talk about index hopping and that stuff we actually talked about yesterday. Let's talk a little bit more about these different kind of features that you can have, what a bad one looks like, and so on. But also thought experiment here. Realize that in this example that we just did, the I1 is enough to distinguish the samples, right? So the I1 is unique here. And this run could have been done as a single index run. You would still be able to distinguish all the samples there, and this could have been submitted as a single index run. But what would have happened in the output data in that case? Well, at that point, I2 just becomes unobservable, right? You no longer distinguish I2. There's just no dimension there to grade on. So what that means in terms of, since we do know what it would look like as a paired matrix, what that really means is that all you get is that you get each row, but all the columns for that row collapse together into one output pile, right? That is what would happen. And so the Demux pile for sample 1 here would be the entire row in this guy, including all this yellow stuff. And you would have 1% of other things flowing in to him, not like 0.1%. So here you would have much more missed Demux, and you wouldn't know because you couldn't observe it and so on. And you don't have this mitigation defense. You're having sort of these decoy empty wells to soak it up. But yeah, we know what would happen exactly in this run if it had been done as a single index guy. It's just collapse this matrix, get rid of it, just stop worrying about the column, just collapse each row into a single square. That would be what the output piles would be. Okay, so these are some of the things you will see. In a good run, you would get whatever your pattern is, and then you would have black in the rest of the matrix. If the color scale, if 0 goes to black, that would be the bottom. Or I think in these color scale, there you see some black there. There's like 1 0 square. Purple, in the rainbow scale, these are all log scale for colors, and black is a 0, a hard 0. Purple is like 1, and then it kind of goes up in logs until you reach red, which is the maximum. Red is where the user libraries are. The top user libraries are the strongest red. And these have all been clustered using like essentially the equivalent to like these p heat maps with the clustering turned on. And so, yeah, so some of these, what you see is something that corresponds exactly to how they were prepped on the wet bench. A lot of people, when they're prepping a lot of libraries, so like you can tell some of these are pretty big plexes, right? I mean, that's at least, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 20, 30, 40, that's like a 40, 50 plex or something. This guy down here is probably more like a 96. It's probably a 96 well plate. I mean, this is probably a 96er. maybe with a couple extra. This little guy in the corner will be 5x or something. Maybe he had a dark poly G in the I2 or something, some extra rows at the bottom or something. But I almost guarantee. I mean, I should know. I made this. I should know which one this is. But this looks like a 96er just from what it's got to be. So oftentimes when people have that many samples preparing, they'll do them in little mini batches, right? You know, you'll do either 8s or 12s or something, and that means you've got the fluids for those guys going on at the same time, and that's, you know, there's a chance there you're going to have crosstalk between those, just because, I mean, if there is going to be some kind of thing going on, it's, you know, it's much more likely to be in those little batches that are sitting together on the bench at the same time than it is a batch that you do the next day or something. And that's exactly what you see here. There's 1, 2, 3, 4, 5, 6. This was prepared in 6 batches, and when we showed this to the guy and he looked up at his notes and stuff, and he says, yeah, that's exactly what I did on, you know, each day or whatever. So what this means is that whatever prep he was doing, there's some leakiness there, right? You know, so there's some free, you know, adapters and molecules are still around or at some point in something, and at some point when they got mixed together, they still were able to kind of attach themselves to free biomaterial and end up essentially mislabeling the biomaterial from, you know, and so on. And so here you in fact in this one you also see that there's kind of these light diagonal lines also. So I think his prep actually did involve little grids, and so he's both doing things with when he does his little mini batches, which are, what did I say, 6s, so they're 96 divided by 6 is what? So it's 16s. He's doing 16s at a time. And I think he might have had those in like little 4x4 grids or something. And it was like, this means that like the rows are bleeding into rows, but not so much the columns. The columns a little, but like the rows a lot or something. And that's how you get this like kind of dual block structure, where sort of like more bleed in one dimension and then these light diagonal lines. No, the light diagonal lines actually cross between the batches. Anyway, there's some structure here that correspond. You know, I don't remember the whole details for this one, but you can see that like sometimes like here's another case where like 2 projects have been mixed, and like this project has some index hopping within itself, and this project, which is a higher representation, fewer numbers of samples project, has index hopping with itself. Both of them more, you know, this one a little less so, but you know, more or less uniformly, but not to each other at all. Like, and so what this means is that the index hopping had to be during prep and not on the instrument, because the instrument doesn't know this, right? The instrument doesn't know that some samples are from one project and some samples are from another. Much more likely this happened. Prep 1 had some crosstalk, prep 2 had some crosstalks, then we mix, but there's nothing going on after that point. So you get a little block here in project 1 and a block here in project 2, but not stuff between the 2 projects. Yeah. I don't know. Our most experience with robot labels is probably from the UNGC core, and I do think we see more hopping from that. Yeah. So I wouldn't be surprised. So if I had to guess right now, the robots are worse. Yeah. Which maybe is the way you would guess. Yeah. Yeah, and nobody, and no eyes, right? There's nothing watching what's happening, or yeah. Like here's an almost completely dense design. The guy left out, you may think this is just a weak library. Like he did a full, what is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is, I think, 2 projects mixed in. This one project is essentially singly indexed. So it has a constant either I1 or I2 here. I don't remember which axis is which, or if I've rotated this matrix by 90 degrees or not. But anyway, one of these projects is essentially single indexed, and the other project looks like a completely dense dual of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 by 3, 30. But actually, this is not a library. This is a square he didn't fill. This is, you know, he'd made 29 out of 30. But but you know, what is that? That's not It's not black, and it's not blue, and it's not purple, it's not blue, it's not even green, it's on the yellow side of green. And presumably, so this is index hopping, but because the design is like dense -1, everybody else has got a little bit at this level. Presumably it's just happening completely in there, but you just wouldn't, you just don't see it. And if you had filled in the last square, we just would have had no clue that it's even happening. Right? So, as I said, also it can happen on the instrument, and then you tend to get just everything to everything, right? So like this one here in this example, this is probably an on instrument one. The reason why it kind of goes from yellow to kind of blue here is because that actually just correlates with this being the highest representation library and this being the lowest. So it just kind of scales with the size of the library. All of the ones I'm showing you here are mostly unique duals, right? Because they've all been permuted so that the diagonals are essentially the user expected pairs. This was intended as a unique dual. This is intended, I think it's a unique dual, but it looks like there's one thing that shares two indices, one thing that shares two indices. I think this was a case where I actually had to add a variant one to capture some a high mismatch systematic one. So these two correspond to the same library, but it's like you needed this extra variant to capture a lot of this library. So it's really still a unique dual design, because in downstream, these two are going to be pooled because they're really the same sample. Obviously, this one at the right is not a unique dual. This is a single index merged with an almost completely dense dual, but the others are almost all unique duals. One thing you can also have is that you can get what looks like index hopping, and you might as well call it index hopping because the effects in downstream is all the same. But this one actually arises from a completely different mechanism. And what's happening in this one is that his targets, the sequences, the design sequences that are used for I1 and I2, they're too close together. And what this means is that the mismatch error that's just inherent in the instrument, like one mismatch, two mismatch, is enough to bridge to the next guy. So what you see are little speckles. You'll see little hot squares in here, off diagonal. This is intended as a unique dual, but off diagonal, you see some hot squares. And those correlate to sequence similarity of the targets. And it's because the error balls around his targets are just big enough to actually bridge the gap to the next guy. So anyway, okay. Here's the worst one I think we ever saw. This was one of our very early NovaSeq runs, a 448 plex in a 2/3 full design. So the brown here is what was expected, and the white should have been empty. But this is what we got. And so, yeah, okay, these are blue, that's good, but that's like orange. And that's a lot, whatever. In fact, if you histogram these, so that green is the expected pairings, red is the unexpected pairings, and we have log representation on the x-axis, they even overlap. And like some of the red gets out into the middle of the distribution of the green. This was there in the white areas, you have 12% of the data, 13% of the data. And for sure, that's a lower bound on the index hopping, because we don't get to observe all the index hopping that's brown to brown. So basically, this was not usable data because the samples are so cross-contaminated here. This is just, you can't do any biology with this result. So when I say it's bad, this is about the worst we've ever seen. But this is enough at the level where you just, almost all experiments would have to throw out the whole thing at this point, because you just can't deal with your samples being cross-contaminated at like the 1/8 or higher level, right? So depending on, some experiments are a lot more sensitive than that. But anyway, so that now you know about index hopping. But try to do unique duals. That's your defense. Okay, we are ready to run BCL2FASTQ2 to actually do the Demux of our paired N. We now know everything there is to know Demux-wise that's going on in this example. So when you compose the sample sheet for BCL2FASTQ2, you have an option. If you can, I generally recommend that you actually demux to the full, what I call the full grid. that means all the squares, all distinct combinations of your I1 targets and I2 targets. And you make each one of those be an entry in the sample sheet, and you have BCL2FASTQ2 produce an output pile for every single one of those. If you have a big dual, you probably want to write a little script to generate those sample sheets at that point, because otherwise it's very repetitive to try to build that by hand. Little Python or R, whatever you like. You got to learn some language to do your little scripts in or something. It doesn't matter which one it is, as long as you know how to use one of them. So for big one, if you're doing a 96 dual, let's call it a 100 unique dual, just because 96 is a lot messier to multiply in your head. So if you have 100 plex and you're doing full grid, how many entries in the sample sheet? 100 times 100, 10,000, right? So you don't want to do that. And in fact, BCL2FASTQ2, the way it works internally, it uses RAM in a stupid way, and the memory usage of it will scale very quickly as the sample sheet grows. You cannot do a 10,000 target Demux with BCL2FASTQ2. Now, there are still ways to get the tiles out in that case if you really want to, but for small cases, the easiest thing to tell you is just have it Demux to the full grid. And so that's what all the purple lines are in here, is that the black lines are the ones that are the user's targets, and the purple lines are the ones that are added as the full grid targets. And then I usually name them kind of like this. So I actually usually put like I number, J number at the front, which is actually the row and column in the full grid matrix. Because if you want to like plot the data or visualize the data or something, it's much easier. You could do it from the sequences or whatever, but this is basically like row number, column number, where it goes in the grid, which is just handy to have. So I embed that into the names. Then I do put the index MERS that are the actual targets for those. It's kind of redundant with the I1J stuff, but whatever. Then I put the sample description, right? So then this could be user sample 1, user sample 2, or something. And then for the unexpected pairs, I usually just put something like unx or something just that says that this pair is unexpected. So something like this. So there's, again, if you're writing a little script to make these, it's not too hard to generate. You may prefer, you do have this project field, which remember that's where the directory that the outputs go into. You may prefer, instead of dumping them all in the same project, you may want to put the unexpecteds into their own directories or something. That may be convenient. So it's just your choice when you compose the sample sheet. The other option is to just demux to the intended pairs. Just the black lines only, but basically. Now that's generally fine, because if you did the R analysis and stuff, you already know what's going on completely, and you've already seen what the counts would be in the full grid. The reason why you may sometimes want the actual reads that go in each of those full grid squares is if you actually want to look in there and see what's going on, right? So if you actually have the reads and you either have more structure on the main reads, like you can figure out something about things from the actual reads themselves or something, maybe you do want the reads, so you can go and kind of actually look and see like what they look like in terms of your data, your experiment or something. But if you're just worried about like determining like what rate index hopping is happening and so on, the R analysis is sufficient, because you've already got the counts. You've already got the size of what all those Demux piles would have been. The only reason to actually form the piles is if you actually want to grab the reads out of there and do something with them and look at them. So like in the Scratch directory, I cleaned it up a little bit relative to yesterday. So I got rid of all the pre-baked versions, and I just made it as if all the commands in the slides would have just succeeded instead of we kind of lost our internet connection yesterday and stuff. But so these are all named to look. This is what it would look like if all the commands on the slides had just worked as written. So I've got two sample sheets here. One is the full grid sample sheet like that, and that looks like that. It's the same what's on the slide, but and here's just the one that just does like the user targets. And of course the user forgot phi-x again, so here's the extra row for phi-x. Okay, in terms of running the actual command, not much changes. Use bases masks. You just have to, like we did with the non-demux dual thing. This is for main end 1, this is for index 1, this is for index 2, this is for main end 2. And here we're just saying I8, I8, which means that when you are gathering sequences to use to decide which Demux pile something goes in, you pay attention to the first 8 bases, which in this case is all the bases of the first index, and the first 8 bases of the second index, which is all of them. So it's Demux will operate on these 16 bases. The decision about where to place the read is these 16 bases. And then, of course, the sample sheet, you just point at the sample sheet. Here is where you put the mismatch tolerance. And it does not give, BCL2FASTQ2 does not give you the option of independently adjusting the I1 and I2 tolerance. This is just applies to both. So this is one mismatch I1, one mismatch I2. And one way to know that's really what it happens is that if you run this, which I did, and so there's this, actually this example and what I ran is with the full grid sample. So we did it with the full grid sample sheet. So then you get that XML file that tells you the size of all the piles, and then you run this XLT prop command that turns that into a text file that just is much easier to use. And that's here. The Demux stats file is here. And if we look at that, that shows these are the sizes of all the piles, right? So of course, so now, you know, remember this column is the one to pay attention to. It's the first one that looks like a count, really. And there they all are. We can actually make that a little easier to see with a quick little, you know, let's cut out column 3 and 6. 3 is this one, 6 is that one. That's the sample name basically and the size. And then we can sort by reverse size. And then we'll just do this little off to put the tally in the first column, and then the sample name, which is longer kind of at the rest. It's nicer to have the number, which is kind of short in the front. And then show that. So we will further take that text file and make it a little prettier to look at here. And so there we go. Now we're looking at the BCL2FASTQ2 full grid Demux sizes of BCL2FASTQ2's outputs. So yeah, at the top is the user samples. Then actually Demux fail is here. And then here's phi X. And then the rest are the unexpecteds. And actually a lot of the unexpecteds, and this is quite common, a lot of the highest index hopping will involve the phi X first index. And then some of it here that's also high is involving that dark I2 for some reason. Well, yeah, so polyg is it phi X or not, right? It's, or if it's just something weird, you know, because hopping means that things are happening with the adapters, right? And so, and dark means not priming, basically. So something's messed up with the adapters. And yeah, so that could really not reflect kind of interaction with phi X so much as the adapters are messed up. Or maybe you have adapter concatenamers, or, you know, I don't know, something, but something's messed up with the adapters. They're not just straight adapter, correct biomaterial adapter. There's something else going on. So yeah, it's often common that you get the darks interacting. That's the most highest, whether you want to call that index hopping or not. It's not going to be in the user section because it's kind of user extra pairs. So, you know, it's less of a problem than user hops. The thing you really don't want is user hops. But yeah, that's a general phenomenon, and it's probably related to the fact that the adapters are messed up in some way to make this happen. You know, for index hopping to happen at all, it's involves some sort of weird adapter stuff that's going on. Oh, so what I was going to say is, if you take these counts and then reconstruct the matrix that we got in R, right? So that's actually straight out of BCL2FASTQ2 with one mismatch tolerance. But this matrix here, which is the one we generated in R, is R, you know, this is only R code make this, and we designed this to be less than or equal to one mismatch, less than or equal to one mismatch. These two views exactly agree to the digit, right? So every single one of these numbers, when you figure out where it goes in the two matrices, it's the exact same number. So it means that our R view is completely accurate in terms of a simulation as to what the BCL2FASTQ is doing. We completely agree on what we're doing in those two worlds. Okay. Okay. All right. So there's a couple more things that are less directly connected to that are less directly connected to demultiplexing per se, but are more related to index reads, because there's UMIs and there's what are called duplicates, and I want to talk about that some. That'll be kind of where we end. And so we don't really have time to kind of get into like the full, like do examples for every kind of possible kind of problem I've seen in the 600 lanes that I've done. And especially once you get into combinations of them. So like some of the problems that happen one at a time, they're not too hard to figure out. But then you get lanes where like the representation ratios are crazy and the user's targets are too close together. And it's a 100-plex mixture of 8 different people's projects and. And when these things all combine, it gets very, very complicated to try to figure out what's going on. So anyway, so just to mention again, kind of at least kind of what problems exist without really giving an example or talking about the combinations of them. Yeah, so a lot of these we've already mentioned, but all right, the user sample sheet could be wrong and could be wrong in a very straightforward way, like I1, I2 reversed, I2 is reverse complemented than what it should be, and so on. There's a fairly discrete numbers of those, and those are kind of like the number one thing you try if there's like the whole lane is like, if you have massive damage in the lane, that's the first thing you kind of try. Then there's the whole class of problems that derive from violating our mixing rules. Stay at least 3 mismatches apart, right? Minimal guideline. Keep your targets at least 3 mismatches apart. And for dual indexing, that means you don't get to concatenate your I1 and I2 and get 16 bases and then have those be 3 mismatches apart. I1 better be 3 mismatches apart just within distinct I1s. I2 better be 3 mismatches apart just within distinct I2s, because index hopping is going to give you all combinations anyway. And so you don't get to have just the fact that you've used certain pairs out of the list to kind of get your distance out of there. No, you got to consider I1 and I2 to stand on their own. Then you've got the whole class of problems, which are basically extra samples and missing samples. It's nice when the number of missing is the number of extra, although that does leave the user with the problem of trying to figure out which ones are which, but at least kind of, it means that the right number of samples is there, which is better than like, missing 8, found 4, missing none, found 12, all kinds of things happen. At high representation ratios, in general, that is the worst compounder. This in the presence of any of the other problems, always a big headache and really complicates interpretations and so on. We talked about the ways that the index base caller can go crazy, so that you can have either low mismatch, low indel corruptions that become high frequency, even beating out the expected sequence. They don't even have to be low mismatch, low indel. They can even be very far away. In fact, one of the things that can happen with phi-x is that if phi-x ends up being a large percentage of the lane, is that there seems to be a tendency for the base caller to start making chimeras, where half of the index reads will come from the phi-x sequence and the other half will be from the user sequence. And that can very quickly produce you sequences that are very far away from the expected thing. Index hopping, of course, and to the extent that hopping has structure, right? One of the things you get from these plots when there's structure is this means that your samples will not be cross-contaminated uniformly. There are little blocks of samples that are intermingling with each other and others that aren't. And this means that if you had some analysis that was, sensitive to some feature, well, you may actually recapitulate this index hopping structure out of your analysis because it actually looks like certain samples are correlated with each other, and it's not because they're biologically correlated, but it's because that's the way you prepared them in batches on the wet bench, and you've actually got index hopping going on in there. So depending on how these blocks cut against your conditions and stuff, various things can happen. Which is why sort of when index hopping happens, you sort of do want to know what the structure is so that you know which samples are commingling kind of with which others. So you know to either kind of be worried or maybe when you do your interpretation of your results, you kind of know, you got some little caveats in your head about what features, you might have to be worried about or something. Yeah, we just talked about a little bit about damage from phi-x. My current recommendation is actually don't spike in phi-x. And any of the cores, when you submit your sample, you can request no phi-x. It's more complicated if multiple of you are mixing and you're each independently communicating with the core. But yeah, anyway, my general recommendation there is these days, lanes don't tend to fail. The instrument tends to be fairly reliable. Flow cells are fairly reliable. There's usually, it's obvious to us what are library failures and what are instrument failures or reagent failures. That use of phi-X isn't really needed anymore. If you really need phi-X because you need to even out your ACGT distribution, find another way to do it. Either prepare more libraries that are not of that type, RNA-seq, genomic, something, or find a friend with libraries like that, mix them together with your lane or something. Don't just pile in half phi-X or something. Anyway, okay, so that brings, so that's sort of the end of the sort of the QC detect and remediate section of the workshop. And let's talk now about these some of these related topics to index reads and not directly Demux so much, but kind of related topics because they involve index reads. And so, well, maybe this is also good. Maybe this is a good point to see if Eloy has sent me his QR code or whatever for the feedback form. It's always a question of which account he sent it to. Oh, here we go. Okay. All right. I'm just going to walk. There's only 4 of you. I'm just going to walk around. You can scan it from my phone to your phone. Okay. Maybe I'll let you do those notes. I got to get some water. I think Eloy has some bottles. So.
That's Sergey drinking. Oh, I've never, but she did.
Oh, they're doing it now.
Oh, you sent it by email.
There's only 4. I just walked the phone around.
Just turn on the slide.
But then I got to get it from my phone to the laptop. There was quite a bit of water in there yesterday.
Yeah, well, I have to draw every Monday and Thursday because over the weekend it's possible. I just think the end Monday first. It's halfway up.
It's better. I mean, it's actually hard to believe that much was going in the wall.
Well, it was, but I don't know. You just kind of see the creases on it.
Yeah, I know, but it's a substantial amount of water to be dumping into, the walls and ceiling.
So
no, that's the DI water.
I don't think so. I just that when I got here on Monday, it's all the way up here.
No, I think the pipe is actually leaking. I mean, if you take the ceiling off, the DI pipe is literally
that's why they
leaking.
They put that. That's what's leaking here. So if you see the drywall, so kind of yeah. So you see that they have the water.
We used to have it come. We had water dripping out of like where the projector screen comes down.
Yeah. During one of our lovely meetings, one of the catering people saw is getting wet.
Yeah, they're like getting wet.
But yeah. it was
Which
like-
means for sure there's got to be mold up there now.
It has to
Super
be.
concerning that they have to
I gotta see what's going on.
They didn't seem too concerned, and they've actually put like a brass fitting into the ceiling now to actually. It looks not so temporary right now. this
They
drain
order-
pipe,
Just
this
put it
drain
this way,
tube.
just put it this way, they order it parts.
I mean. it's
But you just had this whole. Yeah.
They-
You were mentioning the whole like cracks in the pumps takes forever to get a new one. Was that you?
Yeah, it was
Yeah.
cracks and pumps. We also have like legit like practice distributors that operate cold rooms. Like the cold room which will operate the distribution side.
there was something.
Yeah. And they had to reorder like 15, but then they're out of stock.
What's happening?
It's like 2 years. And then they come back in stock to get replaced.
I don't get that. I do think Fairmont is a nice building. There's only like a-
A building can be brand new and have serious problems. Yeah.
But
CSU Dominguez Hills, where my wife is, they have a brand new science building. Ready right at when COVID started. So nobody was in it for 2 years. Well, guess what? The warranty from the builder is 1 year. So the very first thing people do is go back into it and find all these problems. And like the stainless steel sinks in some of the labs rusted. I mean, just weird. I mean, just power keeps going out in this building. There's like arcing in one of the underground vaults or something. I mean, I don't know. It's and it's a brand new building. Brand new doesn't mean no problems.
it also depends on how well they.
Yeah.
They don't have staff to actually take care of stuff. The actual.
So you said it's a cutting market.
No, it's just the continuous battle of man versus entropy. I mean, it's just at some point you'll have a house and you'll understand that
No.
the natural desire of a house is to fall to pieces.
That's what apartments. It's not my problem.
Okay, is that enough time? Everybody's done with the survey. Okay, thank you. All right, so yeah, question? No, the output directory in BCL2FASTQ2, the output directory, if it exists, I think it has to be empty. I think it'll refuse to run in an output directory that already has stuff. If it doesn't exist, it'll just create it. You don't have permission to read the 99 because I don't have permission from the owner of the lane. That's a no permissions to read file thing. Okay, so let's talk about duplicates. Duplicates are what's going to inspire UMIs, and UMIs live in index reads, which is why we're going to talk about them in the Demux workshop, because the Demux workshop is all about index reads. So it's a natural place to talk about it. So a lot of sequencing experiments result in duplicates. And what is a duplicate? It means two wells, in our case, the nano wells, right? The thing that are, I don't want to say read, because the read is like you get, the main end one, main end two, index read one, index read two, those all belong together. And the thing that ties them together is the well. They're all coming out of one well in the sequencing experiment. So it's a well whose reads are the same sequence, basically. Like all of them are, if it's SE, I1, I2, PE, all the same. That's a duplicate. Now, when I say same, generally you got to allow some slop there, because the sequencer makes errors too, like that, there's base call errors and stuff. And what we really mean is that sort of like the DNA in that well is the same as another well. So in the absence of base call error and so on, it would be literally the exact same sequences that out of the reads. But we generally have to allow some slop because we don't get to observe the true DNA in there. We only get to observe what the machine reports. So, allow some mismatches or something to declare that two wells are the same. And the tolerance for that, it depends on a lot of things and how worried you are. Like some people in some experiments are very worried about duplicates, and they would rather throw away stuff that is a chance of being a duplicate at the expense of killing things that really aren't duplicates. So they want to be more conservative. So they would use more slop. And then other people are like, oh, I'm much more concerned about false positives, throwing away data. And so then you use less slop. The other thing that pushes people in the less slop direction is that computationally allowing slop, even moderate or even sometimes just non-zero amounts of slop, can be computationally very expensive. So if you imagine getting a full lane sequencing data, which in an S4 lane is it's like a billion reads or something, right? So you essentially have to do an all versus all comparison there, right? If you haven't done an alignment to a reference genome or something, right? If you just literally just got this gigantic FASTQ pile, and you're going to have to compare every read to every read to like decide which ones are duplicates of each other with slop, right? And so it generally means either very slow or very high RAM to build some fancy index structure or something, but it's not an easy problem at that point. So there's two major approaches to identifying duplicates. One way is if you don't have alignments yet, right? If you're really trying to do it pre-alignment, alignment-free, just on the data out of the lane. The other methods say that, oh, in my pipeline, I'm going to have to align to a reference genome anyway. So why don't you align to the reference genome? The aligner knows how to leave quite a bit of slop there, right? And then we'll only have to worry about finding duplicates nearby each other on the reference genome, right? So that sort of pre-filters it a lot, right? So because the idea is that two duplicates should hopefully land by the aligner almost in the same place on the genome. I mean, maybe a couple of bases on the edges of the reads didn't align and got whatever, but within a few base pairs shifted left or right, it should be basically on the same place on the reference genome. And that means you don't have to do an all versus all problem, just a very, very local comparison in one genome, much easier computationally. So why worry about duplicates at all? And that the main reason is because a lot of analytical pipelines, one of the key assumptions is that every read, every well is an independent observation on the original biomaterial. Right? So, for example, think about like GATK or something where you're doing a variant calling or genotyping or geno calling, either discovery or typing against known like dbSNP or something. Then in human or something, especially if you're doing a large number of samples, you may be doing that with sort of like barely sufficient coverage. That's actually the world that GATK was written for. Like coverage is like average coverage 3, 4, or 5 or something, right? So in human, it's a diploid organism. It's either homozygous allele 1, heterozygous, or homozygous allele 2 in a simple SNP case, where sort of like there's only two alleles at a single point locus, for example. Now, if your coverage is like 3 or 4, how are you going to make that decision? Well, individual reads matter a whole lot at that point, right? I mean, you see like AAC, heck, what else are you going to do, right? I mean, you're not going to really default to the assumption that the C is some base called error of A or something. Right? No, that's going to be a head. AAA is an A, CCC is a C, but pretty much with small numbers of reads, as soon as you see the 2 letters, that's a head, right? I mean, you don't have enough data to cover it. I mean, if you have coverage 20 or something, then it becomes very obvious, right? 0 or 1 C's is the A, and 0 or 1 C's A's is the C, and then if they do see both, it better be something in the middle, right? I mean, according to the binomial distribution or something, right, or whatever. But anyway, until you get enough coverage to actually separate the left edge and the right edge in the middle and see some holes in the middle, then you can't do anything besides basically just call head when you see both. So in that case, duplicates kind of really are worrisome because, if you see AAC, if you see like AAAC, but that A is really 4 copies of one original observation, and it just changes all the calculations and statistics and probably all kinds of things and your error rates and stuff. So, you know, and basically anything that counts is at risk, right? So that's RNA-seq, ChIP-seq, BSCRC, whole genome bisulfite, especially because your coverage tends to be low. Anything that counts is going to be affected in some way by duplicates. Duplicates that are false, right? I mean, some of the duplicates could be real, right? I mean, due to the biology and statistics of sampling and whatever, in some situations, you really will get some reads that are exactly the same, and they represent independent samplings from the original material. Those are real duplicates. Duplicates aren't necessarily automatically bad. They're bad when the amount of them you get out of your experiment differs from what should be there based on the biology, right? That's the thing that's worrisome, is when the rates go out of whack with respect to each other. So when you have a lot of bad duplicates, anything that basically involves counting and so on is going to be affected to some degree. And like things like GATK and whole genome bisulfite are generally more worried about it because their coverage is generally quite low at any given locus. And so the effect of duplicates going from 1 to 5 or 1 to 3 or something may change the balance at that locus a lot, because coverage is only 3 or 4 or 5 or 10 or something anyway. Things like RNA-seq, ChIP-seq, and so on. You know, in RNA-seq, you're usually not worried about the genes at the bottom, right? You know, one coverage 1, 2, 3, 4, 5. And, you know, a low gene is like hundreds of counts or something, and then there's the genes with like thousands, tens of thousands, 100,000s of counts, and there maybe duplicates happen, but as long as the duplicate rate kind of is distributed everywhere. I mean, in RNA-seq, you're averaging over the whole length of the gene anyway, right? I mean, so you know, as long as things are kind of fair, it'll just kind of duplicate whatever the duplicate rate is, as long as it hits everybody about the same. It's sort of okay. I mean, anyway, but yeah. So kind of the sensitivity to this depends upon what you're doing, and some types of experiments and analyses are more sensitive than others. So where do they come from, right? So I mentioned, yeah, some of them are real. The true ones, which green in the slides here. That'll be our color for the good ones. And some protocols like reduced representation bisulfite, RRBS, and even RNA-seq to a degree are actually going to generate huge numbers of those by design. In RRBS, it comes out because you digest the material with a restriction enzyme, and then all of your reads are going to start. They're going to be all your read starts are synchronized to digest locations, and there's a much smaller number of those on the genome than there are whole genome positions. And so you're concentrating all the reads on a much more restricted set of places. And since the methylation is you know, say it's a binary mark, and after bisulfite conversion, the unmethylated cytosines become T's, so the methylated cytosines stay the same as C's. So it's like a couple C's are flipped into T's around the reads, depending on the methylation state. But like a lot of the methylation profile is fairly stable, you know, even cell to cell, individual to individual even. And so since you synchronize the start place of the reads to just these subset of loci, and the methylation state is kind of like, you know, it's got patterns and stuff, and it's not like completely random, you totally expect in the pileup to have real biological duplicates, a lot of them. I mean that's just part of the way it's going to be. And even RNA-seq to some extent, like for example, if you did poly-A enrichment or something, right? Well, you kind of put in a bias towards like the 3' ends of things, right? Because that's where the poly-A tail is. And then some genes are very high expression, you know, you have 1 million copies or something in your 20 million reads or something for like, you know, I don't know, actin or something, right? It's going to be very high. Then you're going to get some duplicates there, right? I mean, just because that's the way the statistics say it should work. And we do not want to throw those away, right? Those we want to keep. The problem is that some are artifacts, and sometimes a lot of them are generated, and a lot of them are artifactual at that point. And one of the big offenders of that is the PCR amplification stage in library preparation, because that's an exponential thing, right? And the problem is that if you have something going on where like the source material is not great quality, or maybe it has a really skewed CG content distribution or something, but you may have certain subsets of your original material that PCR is going to preferentially operate upon. And if that subset is small, or the exponential stretches such that those really get stretched out compared to the rest of the stuff, you can end up post PCR with a total of amount of material that looks good, but which is really super horribly over enriched for some extremely small subset of your original material. We call those low complexity libraries. And so if you randomly sample from your post amplification library, you're really hitting the same source molecule over and over again, because his descendants after PCR are just everywhere. You know, he just outcompeted everybody else. And so when you're random sampling from the post amplification stage and you think you're getting random sampling from the original biomolecules, you're not, right? So that's no good. And in some applications, you might have 10, 20% or more of the reads actually derive from PCR duplication and don't reflect true random sampling of the original material. And since that's, you know, so then it's definitely a concern at that point. I mean, it's usually pretty obvious, you know, because at some point you'll align to a reference genome, you look at a genome browser or something, and you won't see your reads like everywhere. You'll see like little buildings like piled up. You know, it's like, oh, it's like 1,000 reads here and 500, then a gap, and then 500 reads here, and then a big gap. It's not like the coverage is even and everywhere. You just get like these little tall pileups in places. That's a real sign that, you know, looks like a low complexity library piece, you know, some kind of amplification problem or something. So we really want to remove those. Those are red, are bad ones. And what does it mean to remove them? Well, ideally, what it means is that you got, you know, you got n, well, n might be 50. You got 50 wells that are all duplicates of each other, meaning they're all the same sequence. Okay, I mean, if duplicates are a problem, you'll have like this group of 50 and this group of 40 and this group. You got many groups, but let's just talk about one group at a time for a second. So you got these 50 wells that are all calling the same sequence. So n equals 50. So removing duplicates does not mean you kill all 50 of those. It does not mean you kill 49 of them and leave one. What it really means is that you need some estimate m, where m is going to be between 0 and 50, about if the sampling from the original biomaterial was fair, how many reads would I have gotten out at the end? So maybe m is 20. I mean, the problem is it's, you know, very hard to know m, and it's even hard to estimate m, but maybe m is supposed to be 20. You're supposed to get 20 copies, but you got 50 copies. Well, now you should kill 30 copies out of your data, right? Not 50, not 49, not 0, not 1. You should kill 30. So this is that's the ideal situation, and a lot of dupe removing software tools don't even come close to meeting that ideal. Okay, like not even close. And the main problem is that without sort of extra information, essentially what the M estimation problem is, you need to tease apart the real dupes from the fake dupes, right? That's the real, we need to decide red versus green, and the reads don't come labeled. Or maybe they do, that's the idea, right? So without extra information, you can't really tell. And then you're left with trying to make generic estimates or use kind of background knowledge. It's very complicated to come up with an estimate for M if you don't have some extra data. And it's going to be extra data you're going to have to extract from the reads themselves about how to estimate M and know how many of the copies to kill. So the question is, okay, so two things here. We talked about the PCR dupes. There's actually a couple more sources of dupes that are artificial. Let's talk about those. And then we need to go into this problem of how do we separate red versus green. So there are other ways to get artifactual dupes than PCR, although PCR is usually the offender whose magnitude is the highest, and the other problems either are low frequency, or when they happen, you're going to have the PCR duplicates worry anyway. And since the PCR one tends to be much higher magnitude, that's the one that when duplicates are a worry, you're going to be worried about PCR ones, and then the other kinds will kind of be taken care of automatically once you address the PCR problem, as you'll see when we get to how we're going to deal with dupes. So, but let's talk about the other artifactual ways that the dupes can arrive. So in the old pre-patterned flow cell world, where the flow cells lane was literally just a uniform oligo code. You remember the current ones have these little nanowells in a hexagonal pattern, and their lithography created. and completely regular. But in the old world, there was just a uniform coat of oligos. Your DNA landed randomly, and then the cluster grew up in place, and so it's kind of like raindrops, like adding randomly. And so one of the things that the image analysis had to do was just decide which pixels correspond to a spot, and what shape they are, and how big they are, and how much they overlap. Because the main problem there is overlap. There's nothing stopping. Your molecules land, and then you have to polymerize up a cluster, but there's nothing stopping two guys from landing very close to each other. And so then their clusters will kind of grow together and be intermixed and so on. And then you're going to see mixed signal intensity from both of those, which isn't going to be great. And so there's a spot calling segmentation stage of the old image analysis. And one of the failure modes it could have, or not even failure modes, this never happened at we're talking parts per million is what these kinds of things would happen at, is that it would fail to optimally segment a guy. And one of the things it could do sometimes is take kind of an odd shaped cluster, or maybe an unusually large cluster, and call it two clusters. But that's the same guy, and it's going to call the same sequence, and it's not an independent observation, very, very clearly. It's just you've taken something you should have drawn one circle around, and you put two circles around it. So that's called an optical dupe. And it wasn't commonly done, but people knew it happened, and GATK was probably the one to be most worried about it. And they actually have this Picard mark duplicates function, which was created, I think its original birth purpose was to deal with optical dupes. And essentially, how would you deal with that? Well, you already know from day one, tile XY essentially defines a physical coordinate system on the entire flow cell surface. So this problem of optical segmentation, of course, will create two spots whose physical locations on the flow cell are essentially right next to each other. And so you need to essentially, find not only wells that are giving you the same sequence, but if they give you the same sequence and they're physically extremely close to each other on the flow cell, now that looks like an optical dupe. And GATK was and is an alignment-based, reference alignment-based process. So their filters were both, their main filter was walk down the genome in linear order. In your local window around the genome, find the reads that are the same, basically aligning in the same place that are calling the same sequence. Go look at their tile XYs, and if their tile XYs are really close, that's your optical dupes, and you keep one of them out of the little dupe group. Okay. And I mean, that works, but like I say, in our experience, it was never a big deal, because it's like parts per million is the rate that stuff seemed to happen at. And so it just wasn't worth doing a lot of the time. In the modern pattern flow cells, we have XAMP. And remember, the idea of XAMP is you wash low concentration molecules over the surface. Randomly, one of your guys will find himself near the edge or inside a nanowell. He will bind then to the oligos sticking out of that guy, and that initiates the polymerization reaction in the entire well. But that takes a little bit of time, and you're in a fluid, and there's a little moment, when molecules are polymerizing, where there's sort of, they have to move around in that well and stuff. And what can happen, it's not supposed to happen, it's not really supposed to happen, but it can happen, is that a polymerizing well can have one of its member guys escape, right? And then usually what happens in that case is he may be kind of crawling very near the surface. I mean, you have to remember this is a flow cell, so you've got these wells, and you've got bulk fluid. If you go high enough up, you're in the jet stream or something, and the bulk fluid flow is going down there, whatever. But then you've got physics and fluid dynamics going on, and so very near the surface, things are different and slower and so on. There's shear layers, and it's not supposed to be any turbulence, but anyway. But at the very small scale, you've got like Brownian motion and diffusion and stuff, and all kinds of things going on. And anyway, if you get a guy that escapes, a molecule that escapes, mostly what happens is that he will just find a very close by well and start trying to work on that guy, right? So maybe the close by well is already filled, and nothing really happens, because that's just the same as a guy coming in and trying to land in a place that's already filled. But if in the polymerization process he actually gets into a well that hasn't polymerized yet, he can now start a polymerization chain reaction in that guy and kind of fill up. Now you've got two wells that kind of, from your original library material washing in, this guy has now populated two wells, right? And so those are now an artificial duplicate. slightly more rare is that he actually not only gets out of his well, but he gets high enough in the flow stream that he now can take a ride and he can land somewhere further away. It will be in the direction of flow generally. So that first process, he can actually go back against the flow kind of, because the flow isn't really strong down close. But once he gets into the flow, he's kind of biased in the forward direction, if you like. And so you can actually go and look for these. And I did when we first got the HiSeq 4000, which was the first pattern flow cell guide. And you can actually find these, and you actually find that there's sort of like little comet tails. If you sort of, I mean, it's rare events and so on, so you have to average over the whole flow cell and so on, average all the events. But if you sort of decide sort of what is the closest to point of entry point, closest to, most backward position in the fluid flow for each dupe group, and then sort of align all the dupe groups so that those positions are all lined up, and then make a density plot, you actually get like a little comet head and then a little comet tail that kind of comes out. It says that shows you sort of the frequency of where the duplicate is likely to be. And so, locally more isotropic, and then as you get to further distances, comet tail. And it's even when it goes a long distance, it doesn't really go like 3/4 of the way across the flow cell. It's still a fairly short distance, but there can be a lot of wells from here to there, thousands of wells away, but not millions of wells away. So anyway, you can look at these, and if you even do the statistics carefully, looking at like triples and duplicates at a time and so on, you can even convince yourself from the data that the flow, the duplicates really do occur in the direction of flow. You have to, I mean, if you just have 2 dupes at a time, you can't really tell who is first, who's the parent, who's the child. But when you start looking at 3 or more at a time, you can sort of tell, because you know that the 2 that are closer together are probably closer to the point of origin, as opposed to if you got like 2 that are close and 1 that's far away, the 2 that are close are probably closer to where the originating event was, and that sort of then gives you an arrow that tells you which way it tends to go, and then from that you can find out that they all tend to point in one direction, and then that's probably related to the fluid flow. Anyway, I did write like a C++ tool for this like early on, and it uses crazy memory and so on, because essentially you're doing this pre-alignment. It's an all versus all comparison. You can't really optimize for just looking at local places on the flow cell, because it's a pretty wide number of cells where this can happen. And anyway, and you can figure this all out. And the rates that it happened were not high enough to really cause us much worry. And so eventually I just stopped checking. And the reason why you don't really see people look for and correct for those kinds of problems is because if you're worried about that low frequency kind of stuff, optical dupes, XAMPP dupes, PCR dupes, when they happen, are like orders of magnitude more of a frequency. And so in that case, you're going to be doing something to address the PCR dupe problem, and that's going to, as a side effect, end up taking care of these other guys. I mean, so it's the reason why nobody does it is because if you're worried about dupes at all, you're going to have to do something that's going to worry about orders of magnitude more problems than these cause. So since these are kind of hard things to kind of deal with, that's why you don't really see tools that are just dealing with that kind of problem. Okay, so that's sort of all the ways that we can kind of get real ones and fake ones. And now we need to be able to isolate red versus green to know how much to keep in each dupe group. And so one way people do that is to just give up on kind of the fine scale and just use basically a sledgehammer. And that's basically to, in each dupe group, you just keep 1 copy. That's actually not that uncommonly done when people do deduplication. And whether that kind of works well or not depends on a lot of details. But so that doesn't really require a lot of fine-grained handleness. It just means you find, however you do it, reference alignment or whatever, you find all the wells that call the same sequence, and then you just keep 1 copy out of each group, right? That's like a sledgehammer. For sure that, each sequence was at least 1 original biomolecule guide. You shouldn't kill them all, right? You should keep it, there's got to be at least 1 of those that's real. You had to come from somewhere. But the question is it really just 1 guy that blew up into the whole group, or is it really a number of guys that are biologically real that some of them blew up into bigger groups? So you shouldn't kill all n-1, but you should kill something less. So anyway, if you have single end reads, there's not much you can do besides things like that. Paired end reads are much better because when you prepare a library, there is no way that you can do size selection on the fragments before you put the adapters on or after you put the adapter on. It doesn't, you know, whatever. But your user DNA fragments, which the length variation of those turns into length variation of the final molecules that go on the instrument. There's no way when you do the wet bench that you like precisely cut out the guys that are like 127 bases long, like plus minus 0, right? It's more like 130 bases average plus or minus 50 or more in a, like a Gaussian normal distribution or something on the sizes. So if you run like a bioanalyzer or something, right? So then you'll get actually, you know, a tray. It'll tell you like the actual size distribution, right? It'll basically the x-axis will basically be nucleotides and the y-axis will be frequency, and you'll get a little plot something like this or something, right? So this shows you that your molecules have a mode, you know, there's a mode for the size or a mean for the size, but then there's a spread around that size. And when you do paired end sequencing, remember you get the, you know, here's your adapter, here's your insert, here's your adapter. This part in the middle is the thing that's varying the length a little bit. And when you do paired end sequencing, main end 1 comes from there and main end 2 comes from there. And that means that, say, once you align them to the reference genome, here's the reference genome, right? And then this little piece will align somewhere on the reference genome, and this little piece will align somewhere on the reference genome. Now you didn't, you know, assuming that the insert is long enough, you didn't sequence the part in the middle, right? You didn't get to observe this directly from the read. But because once we align it on the reference genome, we know this place and we know this place, and now we can infer the length of what the insert must have been. Okay, so paired end reads, at least in the alignment-based mode, you know the length of the insert, and that's a random variable. That's a randomized object, and so two original biomolecules. If you have an original biomolecule and it goes through artificial duplication, that will be preserved. That link will be preserved because PCR and the XAMP dupes and so on, they can't change the sequence. They can't change this molecule, this you know, whatever. So they will preserve the insert length. But the biological guys, you know, there's no reason to believe that two independent copies of the biology have any preference or not for the length distribution. And so they're just gonna, you have two bio guys, they're just gonna independently randomly draw from the length distribution. And so the chances that they have exactly the same length are small, if there's any variation in the length distribution at all. And so this is great. So the insert length provides sort of like this auxiliary randomization. It does exactly what we want. Artificial duplicates preserve it, and real guys feel the randomness. And so what we can do is align to the reference genome. And then to call two guys a duplicate, not only must the reads have to be the same, but the inferred insert length must be the same. And this is very good because this is the first time you actually have a chance of separating out the reals from the fakes. Now, if the insert length distribution doesn't vary much, or your coverage is extremely high at a locus, you're still going to run into trouble because you don't have enough randomization then. And you're going to start having a non-zero chance that real guys are going to share the same insert length. I mean, this is just a discrete number, right? And it's like, maybe if you have a super tight insert length, maybe you only have like 100 to 120, but it's mostly concentrated at 110. And, you know, and then if you have coverage, hundreds of coverage at a given locus, now there's non-negligible chances you're going to get multiple guys that exactly hit 109, 110, 111, and now you don't totally get separation between truths and false and fakes anymore. So it's not ideal, but it's better than the sledgehammer. And so this is pretty commonly done. It is with paired end reads, people's dedupe, what they will do is find all the reads that where the start and ending location, the starting location of main end 1 and the ending location of main end 2 are precisely the same. That's a dupe group, and people will just keep exactly one read out of the dupe group. That's a common approach. And that works well until you get to the point where the randomization and the insert length in your coverage is such that you no longer have a high chance of real guys being different in insert length. So, okay. But that brings in essentially the essential idea to solve the problem more better. And that is introduce randomness. Introduce randomness that the reals will feel, but the fakes won't. We just don't have enough with insert length. So we need to introduce more randomness. And where can we introduce some randomness? And one answer is, since index reads are so long these days, we will just throw in some random bases somewhere in the index read. That can act as our auxiliary randomization. And those are called unique molecular identifiers, or UMIs. So your UMI might be in index 1. So here's the adapter for the left side, the main end one side, there's the adapter. Somewhere in there is the little variable part that you sequence to tell what Demux pile to put something into. Well, we just make that a little wider, and then the back end of it or something is just random nucleotides. When you order oligos or whatever, you can specify just N, right? And then the chemistry guys are supposed to, instead of carefully making sure that only a single nucleotide goes in there, they just use a randomized mix, and then what gets incorporated is just a random choice by whatever chemistry and physics decides. And so the idea is that your final prepared adapter mix, that goes into one tube, that goes into one library, one of your samples, the first part of it is completely stable where the index is that you demux on, but then that variable portion is ideally just completely randomized, right? If it's 8 nucleotides, then you've got 4 to the 8th power. How many 8 mers are there? Well, 4 to the 8th power, which is the same as 2 to the 16th power, which is 65,536. So, usually these syntheses for oligos, they're not perfectly random, right? You'll get some whatever, but it doesn't have to be perfect to be useful, right? All it's got to do is introduce randomness so that the bio guys feel the randomness and that the fake guys don't feel the randomness. The fake guys not feeling the randomness is automatic. It's just more randomness is better. So whatever randomness you got, that's what you got. And if it's bigger, that's better. But any amount of randomness is helping. So that's UMIs. Yeah. So let me just mention that you may see tools like FastQC or FastP that you might have used for quality control on main reads and stuff. Sometimes those will report numbers that are duplicates or repetitive. You got to be real careful what that means, because they're usually very unsophisticated, and the interpretation that may just not really make any sense at all. And they don't know anything about UMIs or the paired end insert lengths or anything. And so they are estimates that are kind of cheated in various ways. And in general, I don't even recommend paying too much attention to those, because it's just too disconnected from reality. Right. Okay. So we've talked about all of this. Yes. Okay. So it's kind of beyond our scope to kind of get into the actual way you apply software to do the deduplication operation. And it kind of depends on what your downstream pipeline is and so on. And some pipelines have it built in and stuff, and they're expecting the UMIs to be in certain places or whatever. But from the point of view of the Demux workshop, our main goal is, since Demux is the place where we go from the raw run folder to the FASTQ files that go into pipelines, if we don't if we don't have the UMIs in one of our output files, the pipeline's not going to have access to them, right? If the UMIs are embedded into the index reads, it's our job to at least report them in the files so that the next people can actually use them, because we don't know how to use them in Demux. That's like a pipeline detail or something about how to do the deduplication. So I don't really want to talk about the software that you use to actually affect the deduplication itself, but I do want to talk about just us getting the UMI bases reported in some files so that you could pass them on to the next stage. And there are multiple ways to format the UMIs, like whether they're an independent FASTQ file or whether they're sort of embedded into the read names somehow or something. And different software depends and so on. But the idea is that, format transformations are more or less easy, right? There's the joke that what's molecular biology? It's basically the endless transfer of one clear liquid from one bottle to another clear bottle, right? One pipette, one micro centrifuge, one centrifuge tube to another, right? You're just always pipetting around clear liquids to clear liquids, right? In bioinformatics, the equivalent joke is basically all you do is text file format conversions, right? That's basically what half of bioinformatics is, at least. So yeah, so as long as you get the data in some form, you can always transform it into whatever particular way that the downstream software wants. But if you don't even have the data, you're stuck. So we actually talked about this, I think, on day one, where I said that, like, if you do have UMIs, when you do the non-Demux run, you can just leave them as part of the index reads or whatever. But when you do the Demux run, you treat them as a virtual additional main end, right? So we, in that use basis mask argument to BCL2FASTQ, you change it from like I16 or something, which would be an 8+8 guy tag plus UMI. You would change it to I8Y8, so that that first 8 gets still used to do the Demux decision, but then the next 8 is treated as a virtual main end at that point and gets its own output file, and that's where the UMI bases go. And so then you have the R1 files and the R3 files for your main ends, and then you have the R2 file where the UMIs show up. And then you have the UMIs, and then you can reformat them into whatever other form the software needs or something, but at least you've got them. So that's all I wanted to say. The particular formats that we have seen, and the first two are common, the others somewhat rare, are we've seen cases where the second index is completely UMI, and like even like 24 bases. The other common one is that the first index is 16 bases, where the head is the tag and the tail is the UMI. That's another very common one. We've also seen 19-mer I1s where it's 8+11, and the 11 is the UMI. And we've seen 17-mer I2s where it's 8+9 for the UMI. And we've seen 17-mer I1s where it's 8 for the tag and 9 for the UMI. But you know, whatever. I mean, it's whatever the library. It's whatever the prep. You know, the library preparation protocol designer kind of wants to do. These, like I said, these days we got 36 cycles or so to distribute to I1 and I2. So that, you know, that can be 18+18 or 36+0 or whatever you want to do. And you can partition those bases between tags and UMIs however you like, and so on. And it's just whatever the person that did the protocol design thought was best. In terms of how you'll sometimes see UMIs expressed in terms of, like, you know, sometimes you get like the pre-sample sheet out of like a kit protocol or something. They'll have a table which kind of lists the index, library number 1, 2, 3, 4, whatever they're describing their kit, and it's, these are the sequences you're supposed to put into the sample sheet or something. Sometimes when there's UMIs around, you'll see them do stuff like this, where the end positions mean that that's where the UMI is. It's more just a communications device than anything else. You generally do not actually literally put in the sample sheet the ends and so on, because you would really just tell use basis mask. Oh, in this kit, what is this? This is an 8+11. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8, 9. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 8 plus, yeah, okay, 8+11. So you would say Y8, you would say I8, Y11, and then BCL2FASTQ2 knows that it's not supposed to pay attention to these to make the Demux decision on, and so these don't appear in the sample sheet because the sample sheet needs to have this, or this is in the I1 position, and then this is in the I2 position in the sample sheet. These ends don't literally go into the sample sheet, but you'll often see them presented this way in tables because it kind of tells, because at least you can kind of tell where they are, right? I mean, if it was like this, it tells you that, oh, the UMI is at the front, right? It kind of tells you where it is. You also, it's conceivable, you actually have UMIs that are in both places, right? So you could have, you could have something like that or whatever. It doesn't matter. You would probably, in terms of the UMI output, you would either have 2 files, or you would concatenate these or something. But it's, you could have the UMI in pieces. It doesn't have to be one just sort of stretch. Okay. And I think we also talked about this on day 1 because you had the question, which is what to do when you sort of mix index formats, right? So maybe you have a kit that is sort of nominally singly indexed, and you have another kit that's sort of dual indexed, but you want to mix these on the same lane. Or maybe you've got dual index, and this other guy has dual index, but he has UMI bases and you don't, and so on. So you kind of get these mixed lanes where sort of like, they don't, if they were on their own lanes independently, they wouldn't have been run with the same length of indices, and they wouldn't have had the use basis mask be the same for them. So how do you deal with these? Obviously, the physical run the instrument for the entire flow cell can only be configured with a fixed number of SE, I1, I2, and PE bases. So when we do have mixed situations, it's really just the same thing as what happens when you mix multiple lanes on one flow cell. Physically, we have to round everything up so that the data is collected. So SE has to be run at least as long as the longest member. I1 has to be run at least as long as the longest member. I2 has to run at least as long as the longest member. PE has to run at least as long as the longest member. So at least we have the data to, maybe some things got kind of extra data collected, but we have the data to decide everybody at least. So that's an obvious requirement. And then the way some people handle the mixed situation is by essentially running base or BCL2FASTQ2 Demux multiple times. They will lie about the sample sheet, give it only the subset, and then configure it such that it pays attention to the right bases, but they'll run the single index libraries with a single index sample sheet and tell, ignore these other bases you don't really know, really they're there, but the data don't pay attention to them and so on, and they'll run the Demux multiple times. So there are cases when that's okay, there are cases when that's not ideal and so on. In general, since I've got the bottom-up approach, I usually handle it differently. And instead, you do what I call either lifting or altering. And in lifting, what that means is that you would have, maybe you have a single index set of libraries mixed in with a dual index set of libraries. Like we've been talking about, the instrument doesn't know anything. The instrument does sequencing primers and generates sequence, right? You will get some sequence for the single index libraries in the second index. The question is, what is it? Is it unstable? Is it stable? What is it? And so on. If it was unstable, that'd be less good, right? Because then what should you put in the sample sheet? Anything fixed thing you put in there would not potentially have very good recovery, right? So that wouldn't be good. And in the days before two-color chemistry, that was something of a concern. Because, for example, if the single index libraries really don't even prime in I2, then you're going to get black. In two-color chemistry, black is reliably poly G. And four-color chemistry, black is whatever the base color wants to do, because it basically looks like noise. Base color is going to stare in there, and the camera is going to get random Poisson fluctuations, and the base color is going to try to split this 0, 1, 2, comma 0, 1, 2, comma 0, 1, 2, comma 0, 1, 2, red, green, blue, purple color that the camera is measured, and try to figure out which of those is HCGT and so on, and it'll just make. And then usually the background correction has biases and stuff, and you'll tend to get low complexity sequences out of that, but they won't be stable. And so then that's not so good. But now we're in the two-color chemistry world. Black is poly G reliably. That's not really a thing that happens so much anymore. So even in the worst case where it wouldn't even prime, that'll at least come out as poly G. So the stability question is not really a concern. And so they will sequence as something stable, basically. Either they won't prime, or they will prime to an unexpected place in the adapter, and then you just get whatever sequences are, whatever bases are there in the adapter. And most adapters, unless they specifically have UMI sections that are designed, nobody's adapters have like randomly variable parts. Like only UMIs are designed to have variable parts that others don't. So yeah, so you never see unstable stuff basically, even if you run a nominally singly index case on dual index. And so the bottom-up approach just lets you discover what those single index guys are doing in the paired end world. And typically what will happen is that they will just have some fixed sequence in I2. And then you just compose a sample sheet, this dual index sample sheet, and you now know what to fill in for the I2 for your single index guys. And you just do them as a single job. Now, what happens if you then realize that some of your single index guys end up being very close sequence-wise to your paired end guys? Should have pre-flighted that, right? That's one thing you were supposed to check. How do you check if you don't know what I2 is before you empirically run the lane? Well, if you're in that situation, that's where you ask us sequencing guides, because we know, oh, you're using this kit. We run that before. We know what comes out of I2. Just because you close your eyes and don't check before, it doesn't mean you're not going to cause a collision, right? And one of the disadvantages of the do it separately approach is there may be collisions that you don't know about, and then you're going to get reads that show up in both piles and are partially material from the other side and so on. And just not good. You want to do everything in the unified world where you see all the data at the same time and check everything in the unified world and so on. So anyway, when I say lift, that's what I mean. So basically means that if you have a bunch of mixed formats in one lane, you're going to demux it as sort of the highest I1 that was ever run. That makes sense for any of those guys. and then the highest I2 length that makes sense for any of those guys. And then you're going to try to make a sample sheet that basically has a complete expansion of them to that full length for all components. Okay. And then if there's UMIs involved, like suppose you have I2 is completely UMI, say, and I1 is sufficient to distinguish all the samples. But only some of the samples have UMIs. Well, in that case, the UMIs are no big deal. You're going to have I2 sequences for the guys that don't have UMIs, and then you're just going to ignore that output file in downstream. But your other guys are you're going to keep that file and use it. What if the UMI bases are mixed in one of the indices? So actually, suppose the I1 is 8 plus 8 UMI for some of the guys, but it's actually 16 tag for another set of the guys. Well, you don't actually then demux those 16 guys with their full 16. Those UMI guys, those UMIs are randomized and could be hitting the stuff over here. You know, maybe you could argue not a lot or something, but it's just not a position you really want to be in. So in this situation, what you would hope is that the lead 8 bases are enough to distinguish all the samples, including the guys that are designed at 16. But hopefully they're okay at 8, and hopefully they're still far apart at 8. And then you would actually do the demux decision based on the 8, and then leave the tail 8 as purely UMIs considered for everybody. They're going to have a very biased distribution in the cases where there used to be 16 tags for those guys. But kind of once the UMI bases are sort of intersecting somebody else's tag, kind of makes them less nice as tags. So you kind of either avoid doing that or try to work without using those as the distinguishing feature. So I think that latter case is what I call an alter. So lift basically means making things that make the decision more specific by making them longer. Alter is actually making things less specific by paying attention to less bases. Usually the only motivation for that is UMIs coming in and poking themselves in and kind of trying to use the same base for both the tag and a UMI makes less sense. And ideally you can just not pay attention to the base at that point for the demux pile decision. Okay. All right, so we've got 45 minutes left. Let's take another short break, and then we'll do the last thing, which is to look at some common kits and how they're not always well designed. And this is why mixing is sort of generically causes problems. You really do need to plan mixes because there's just too many conflicts out there. Then we'll do our summary slide of all the do's and don'ts. And then we'll talk about a little bit about sort of what ultimate indexing might be. We have the technical ability at this point to essentially solve all the problems we've talked about. And if we could sort of magically alter all library preps to sort of take advantage of what we can now do, we could make almost all these problems go away automatically. And this whole workshop could disappear. So I can say kind of what that world would look like and how we would get there, but then of course it's not going to happen. But it does mean though that if you're running custom indices, if you're ever in a position where you get a chance to design the indices, you can do much better than the current situation. So we'll take a short break. Yeah, but that's a detail of just how the biomaterial gets associated with the UMI. Yeah. So that is, they have arguably as a salvage, well, usually UMIs are not strong enough to uniquely identify the molecule from the entire region, because that would require quite long UMIs whose randomness problem is quite strong. So, but because most deduplication is done with reference to line code, you do not have to defend the entire lane worth of guys. You only have to defend yourself against duplicates of the land near each other. That's a much smaller. So it is not really required to guarantee that every module gets the literally unique, lane-wide unique. No. That's why even like a 4-based UMI would generally be enough. Because the size of the pile that you can compare with each other is just going to be what lanes very close to each other on, it's essentially just your pile coverage. What a typical pile looks like. What's the worst pile you're going to get? For most protocols you can figure that out, and it's like it's either going to be like single digits, tens, hundreds, or something like that. Not really tens. So that means the UMI doesn't have to be that unique or even that random. Just have to have some contribution to randomness that just the insert length. The insert length alone can be pretty good. But insert length alone with just even a little more randomness is most effective. And any extra randomness you might imply is great, but it's not like you need like 40 bit entries out of it.
So are these kind of issues?
Yeah, some of those are nice. Well, the UMI has to be done as part of the prep protocol. It has to be designed in. And so,
So the user sees this.
as a user, if you're not the one creating the reagents that get made from, then you're not the guy that designs the UMI or controls the sizes or anything about. It's the protocol designer on that side. Are there UMIs or not? How big are they? What are the biochemical? What are the actual details of chemically how they're going to make the biomolecule? That's all protocol development job. All we can do is protocol user.
I used to use the Apple literary design, and I was looking at the library of this.
Well, I mean, these days the owner platform is very well understood, and you can order custom adapters. Right? So even if even many kits, where you wouldn't want to replace all the protocol steps, you know enough to sort of know what you would have to synthesize in the adapter to sort of plug it into the protocol. And then you can order from all of those adapters. You can arrange to either extend the length of the indices or randomize indices after and whatever, and that can be yeah. So there are cases where you can sort of like retroactively incorporate UMIs into the protocol design, but essentially what you're doing is developing the protocol. The protocol that's almost the same as the whole protocol. It is sort of like a revision of the patching. It's really alternate. But yeah, you can. The essential parts of adapters are very well known for what you need to make them work with the flow cell in terms of interfacing with that flow cell is well known. And then of course, depending on the details of how your biomolecule comes to be and so on, there's more requirements in the adapters that they can interface with the biomolecular attachment process correctly. But whatever, you have all these constraints, some of the constraints from the platform, some of the constraints from the biomaterial process, and then to the extent that you have flexibility left, you can get UMIs in the place of your flexibility left, and then you can have somebody custom synthesize it, whatever.
How about the spike?
Well, it's in the details. We generally do not want. You don't want the intention spiking anything that you need to Demux with, and then have the Demux output how needed. So that you don't want to do, because from what we talked about, the reads of any small Demux file output are always. You competition against spiking. Don't spike in anything for which you care about the output. Right? If you spike in a small amount, and even if it's other libraries, a small amount, even if all of it goes up. So it shouldn't harm other things unless its index tag does kind of, I mean, if its index tag is close to something else, then you're in a situation where if you try to do Demux and properly separate out those things, you may end up having a problem with Demux tolerance not as low and probably always can suffer. But like I said, if the thing you spike in is really low concentration, you could just give up and trying to isolate it, let it leak into whatever it's going to leak into, leave it off and it's not completely pretend it doesn't exist, separate, or whatever. That's great. But the actual concentration is low, and in moderate size or large size outside, it would be a very fraction. But I mean, you know, there's really, I mean, so I mean, basically, but then what's left to why spike it in? I mean, it's kind of like, what is it going to do? So it's at what stage is this spiking supposed to produce meaningful information? Because it can't be after Demux. Then it causes problems. So I don't know if you're spiking at something that during quantification is what that should matter. And maybe it's okay, but then why not just arrange for it to have adapters that don't participate in XRAM or something because they participate in Demux? Basically, you shouldn't have any spikings that survive the sequence. They generally just cause more trouble. Unless they, I mean, if they're very low concentration and their indices really are far apart from the region, yeah, I guess okay. But I still wouldn't try to take any interpretation of data into account. So what I was
Oh, but this looks like a spiking that you apply even for it. This looks like additional raw material. This doesn't even have adapters or indices or anything. This would be something you would put in, and it just acts as additional biomaterial into your sample. That's not a spiking in the sense that I'm talking about spiking. That is not going to look like a separate library. That is something you're going to put into. You can have, it's just something you're going to mix in the biomaterial. So it's really, you're just having biomaterial that's got more additional stuff in it. But then you would go on to the adapters on and so on. So in terms of separating this out, this would just sequence out with your other biomaterial. Its indices will share in your main biomaterial. This plays no role at the level of the output. It's just going to come out mixed in with whatever sample you put it in. So this is not a spike in the sense that people talk about. This is not something that looks like a separate library. Here you're just mixing in some more biomaterial before you even put the identity of the biomaterial on the bottom. So you're going to tease it back out by aligning it to, you're going to rely on essentially the main read to separate it. That's what makes it. That this is not going to be separated out based on here. It's going to be separated out based on the. So it causes less problems because it's not going to interact with Demux at all.
That's actually an example of, in a way, you can think of that as an example of the other way of doing indexing that we haven't talked about at all, and which is rather uncommon now, which is to actually embed the sequence that determines what Demux file it goes into the main read, not into the index read. That's what people had to do before there were index reads, of course.
That's super old.
That's super old, yeah. Yeah, because at the beginning when we had the first platform, the GA1 or whatever, it was this magical box, and you could wish for it to do all kinds of things that it doesn't come out of the factory or with the kids to do, and you just had to work your mind into some way that you could just. I mean, what the thing does is sequence, right? And you didn't even have paired ends then, right? You only got one sequence. So anything you wanted to do that you could embed into the sequence that it sequenced, that was your way to get information to it. So if you wanted to multiplex samples, well, you got to arrange for them to somehow show up in the main read, because that was the only read you got. And what was the way to do that? The answer is to synthesize custom adapters
But instead of having the sequencing priming place be right at the end of the adapter that goes immediately into the molecule that gets joined to it, that's your insert, it just has the little index tag sticking out right there. So the adapter just comes pre-made with, another 6 or 8 bases, which just, from the sequencer's point of view, that just is like the beginning of the user's insert. But the sequencer doesn't know that the ligation occurred 8 bases later. And so that was how you did multiplexing before there was index reads, right? So you just had tubes of adapters with just, the index read essentially already ligated on where the user insert would go. And now the end of the tag is now the ligation point for the user's insert. That's how we did it. And in a way, what you're talking about is kind of like that. Well, here you don't even have like the adapter sequence on the index sequence on the front. It's just that the main read being 50 bases or 100 bases, and whatever sequence this control is, it's just so different from human, mouse, or whatever. It basically doesn't match any of the anything from nature. Then we can just we just throw that in as an additional component for our reference genome, and then we align. They'll all get sucked into that one place because they're not sequence homologous to anything in the page. So effectively, it kind of self-tags, declares it. It's so sequence-wise different from nature that it just the sequence itself already acts as a tag.
Yeah.
Oh, yeah, that's what oh, you're right. Oh, and then Eli must have taken. Hey, just see Eli before you. You're right.
Okay, so let's do the last few slides here. And I mentioned, I think on day one, that commercial kits, even if you restrict to Illumina, say, as a manufacturer, now Illumina has multiple product lines, so you may think, oh, okay, so suppose you restrict to Illumina and a single product line, like one system, like one kit system or whatever. That's surely got to be compatible within itself. No, it's not. I'll show you. Okay, so remember we've got our little index checker web tool here. So let me load that up and we can poke sequences into that to check. But here's a bunch of sequences. Just to keep the size of this grid from blowing up and being filled with a lot of white space, let me just restrict it to the cases where there's some conflicts so that it's more easy to see. But here, for example, are TrueSeq 4, 6, 11, 12, 14, 15, 18, 21, 25. That's Illumina's first system. That was like their main, like when they first introduced indexing, it's the TrueSeq system. 1 to 12 was the first box, and then it kind of got to 1 to 48. Well, maybe there was a 1 to 24 first. There's 2 boxes, and then there was up to 48. Whatever, but this is their first system. So here's 1, 2, 3, 4, 5, 6, 7, 8. There's like 9 indices from there. Then they have their UDI system, their unique dual indices system. That is Illumina's version of unique dual. And here are a bunch of those from that box. I think that's a 96 kit. Then there's Nextera, and then that kind of got absorbed into Illumina. There's the Nextera non-unique dual set. So that comes in a set of I7s and a set of I5s. They have an I1 set and an I2 set, and then you do all combinations of those, so you can do up to dense duals if you want. So there's the Nextera 7s should be the I7s. So these are first index. And TrueSeq is only first index. That was a single index. Here, the UDIs, we're talking about the UDI I1s here. The UDIs have an I2 as well, but the things we're comparing in this group are all I1s. This is I1 only, this is I1 plus I2, but this is the sequences we're comparing here are the I1 half of these UDIs. And for the Nextera non-unique duals, these are the 7 series. So this is an I7, these are all the I1s, okay. So let's, and here's the list of them, okay. So here's the names here. Index checker doesn't want the names, so this column is the actual sequences. And you can go look up on the Illumina website or so on and verify. I'm not lying here. These really are the sequences that I'm saying that they are. But I can copy out that list and we'll go to index checker. And what do you see? Well, there's even a 1 mismatch there and some 2 mismatches here and so on. But, look at that. So here it is visualized in a more compact format where a white square means at least 3 mismatches apart. Orange is a 2 mismatch apart and red is a 1. And so at least in the example I've shown you here, within each system, there's no conflict, right? So if you say TrueSeq only, it's a white. It's The gray is the main diagonal. So obviously everybody's zero mismatched themselves. The second box, which is for the UDI system, we don't see anything less than 3 mismatches just within that. And same for the Nextera. But in the cross ones, we see all these things, right? So here we've restricted to the same manufacturer, multiple kit systems from them. But if you're mixing with other guys, you can't, oh, I'm using TrueSeq and you're using Nextera. That is not sufficient to make you separate. Okay, you got to check the details of which indices you guys are planning to mix. All right. Here is some more from Illumina. Let's look at some I2s. Some I2s is if you're running the current chemistry on NovaSeq, the V1.5 chemistry on NovaSeq. So here are some of the I2s from the UDI system. So here's some more of the unique dual system. Now we're looking at the I2 halves. Here's some of the Nexteras. Now this is the Nextera 5 series. These are the second index of Nexteras. And here is actually, if you take any of the TrueSeq libraries, which are like nominally singly indexed, and you run them in a dual index mode and you look at the I2, they do the same thing that PHYX does in I2. There's a particular point in the adapter where the prime goes, and then you get this fixed sequence. And so this sequence, which starts AGA, TC, TCG, once you've seen it a lot, you actually just recognize it on site. That's common to every TrueSeq libraries with sequences that way as I2. It's what PHYX sequences as I2. And in fact, what a lot of custom, other manufacturers, single index kits, or what you find in the literature as like literature described protocols, a lot of those that are single index protocols run in dual mode would run with this sequence as the I2. Very commonly seen as an I2 for a kit that wasn't designed for I2. So let's look at these. Okay, so we'll plug these in. And okay, we get some more conflicts. Well, let's look at the pattern of conflicts now. Now, actually, if you look in the UDI system itself, there's 2 mismatches apart just within the Illumina I2 UDI. I mean, why would you do this? I mean, I really don't understand the logic here, but the conflicts are UDI 23 and UDI 56 version 2, which is the current kit. And they're 2 mismatches apart. And UDI 24 and 55 V2 are also 2 mismatches apart. In fact, whatever this version 1, version 2 thing was introduced the problem. And I really don't know what the story is behind why these V2s came to be. Maybe manufacturing did, I just do not know. Yeah, they created this problem. So, so this is what I mean. You really got to be careful. You can't even assume that a single system, a single manufacturer's single product line, Illumina UDI said, no, these 2 indices should never be present in the same. You should never use, you should take the tube, the system tubes, and you should throw 2 of these away, right? You need to throw away V6, V2, and 55 V2. Right? You just open the cellophane and toss the tubes, right? I mean, it's crazy. Furthermore, you might think that the commercial systems would at least be sort of like close to the mathematically optimal that you could do. I mean, why not, right? I mean, why wouldn't you put the effort in to sort of optimize things? Well, no, they're not even close. So like I've done some combinatorial optimization to try to explore, like if you restrict to a certain length of index MER and you want a certain minimum distance, how big can you make the plexity pool under those constraints? And actually with 8 MERs and 3 mismatches, which is a higher constraint than you're seeing here. Like, the UDI pool is like failing, they've got 2 mismatches and they're a 96 plex, right? So this is a higher mismatch tolerance, but I can find with the same index length, and I can find 1,330 of those that stay mutually 3 mismatches apart. And actually with 3 mismatches, I don't even need 8 bases to do that. With 6 MERs, I can keep them 3 mismatches apart and still give you 164. And if you're going to give me 8 MERs, then either I can increase the 3, or I can actually start to do things like keep them indel apart, right? I can actually start paying attention to indels and force them to stay far apart with an indel allowed in the mix. So yeah, so the point is that they're not even close to like, they don't even meet like the minimum criterion of like at least 3 mismatches apart, and they're nowhere close to being mathematically optimized. Yeah, like here's the entire Illumina UDI system, all 96 of them. And you know, we can, if you know, you're like, if we just take like the I2s and just pour the whole thing into the Demux checker. It's got to think for a little bit there. This is the biggest matrix we've seen it produce there. There's actually 100 entries here because there's 96, but then some of them have version 1, version 2. So I put both version 1, version 2 here. So there's actually a list of 100. And you can see it's not like it's saturated with 2 mismatcher guys, but you know, they are there. So it's not like at least like full of them, but it shouldn't have any of them is the point, right? I mean, so you can just get unlucky. I mean, presumably, I mean, a lot of people would run that as a fairly dense design, right? You would want to use possibly all 96 in one project or something, right? I mean, it's a nice number. 96 is a plate, right? So, I mean, the fact that, you know, some of these pairs are incompatible, it's just inexcusable. Yeah, so yeah, you can, you know, custom can do better, basically. Yeah, I just talked about this. Here's, I'll actually exhibit one of them just to say I'm not just, you know, making it up. Here are 164 6mers, and you have to trust me that they're all distinct, or you can easily check an R or something that they're all distinct. They're all distinct. Okay, I'm not repeating one of them twice. Actually, index checker would tell us if there was copies. If I put the same one in here twice, it's going to tell us that's a 0 apart. You've actually got a literal duplicate in your list. So let me copy this. There it is. And it is thinking 164 6mers, minimum distance 3. So there you go. That's certainly possible. Compare that to TrueSeq 1 to 48. All right, so here's TrueSeq. TrueSeq is 6mers. And this is 1 to 48. Should be in that order in this list. So that is the same length. And what minimum distance does it achieve? 2. So that's a way shorter list with a worst tolerance. This is the 2 grids to scale if we were to plot them to scale. So, you know, clearly how much bigger this one is. So yeah, so if you ever have a chance where you can develop a custom protocol, and when you do so, you will have the chance to choose your indices because you're going to order, you're going to order some oligos. Consult an expert first, and we can do better.
So back in the day, we did do something.
Yeah, so there's, yeah, so I mentioned there's sort of like, there are, where is it? Yeah, there are other desiderata here that you could possibly worry about. You may want things like balanced CG content, no low complexity substrings, no reverse complements to each other, or certain other sequences you should avoid. Like you have to avoid phi-X probably, and poly-G, and a couple things. But the sets I can generate are so large, you can just kind of empirically try. If you're going to really commercialize a kit, you could empirically try them and throw away the ones that don't empirically work well. There's just you can just generate so many that it's no problem to find large subsets that work very, very well. And some that can also be the synthesized guys don't always get it perfect either. Yeah, that's perfectly fine because 164 is sort of an odd number. So if you wanted to go down to 96, you can throw away like a third of this thing, and so you just throw away the worst third by whatever criteria you like. Anyway, yeah. So here's the promised slide on the do's and don'ts. So if you're going to take anything away from this workshop, it's this slide, right? And maybe show the slide to your friends and stuff and whatever. So these are things that you can either think about ahead of time or whatever. This is stuff that generally do this before you run an experiment, and then hopefully most of all the trouble doesn't even occur. This is, so yeah, number one, no high representation variation intentionally, right? Don't do that. And try to do a good job at making and quantifying and mixing things so that you don't unintentionally create high variation. Don't create low index read diversity. So when people talk about index diversity, It doesn't mean just make a list of sample 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And those sequences as 10 sequences have to be diverse, high ACGT differences and so on. Those get weighted by the representation. And so it's very common in lanes, especially when mixing multiple fields projects. Some of those become 10 times, 50 times other guys. And it's the balance after they're weighted by representation that counts. And so that makes it a harder problem to create diversity because some of them vote a lot heavier than others. And it means also avoid low plexity then. That's another thing. In general, it's better to run, like if you had 100 libraries to run, it's better to run all, and you needed like a lot of coverage. It is better to run 100 mix, multiple lanes. Do not break that into like 10 mixes or 5 mixes that you completely commit to one lane and only run once. For this reason, and also because if you had to do some adjustments and you're going to commit to multiple lanes anyway, at least you got to see everybody once on round 1 and you can make the tweaks round 2 while you still have a chance. If you're only going to run each library once, you have no chance to correct. So we generally recommend, even if it's not going to be sufficient coverage for you, do higher plexity and run it more if you need to run it more. But that's better than running low plexities just once. Yeah, pre-flight and check that your final mixes are at least 3 mismatches apart on your targets. Very, very important. And you might as well assume that phi-x is always present, I2 poly G is always present, and poly A in both I1 and I2 is always present. Those are commonly seen, and so you need to avoid them anyway. When you're doing dual indexing, do not go beyond unique dual. I mean, we still get rather dense duals once in a while, and it's just, you're not really doing anything. Just don't. But you know, whatever. I mean, sometimes you're either not in a position to change the protocol, or you know, whatever. I mean, you know, any protocol is fairly easy to go back to the start and kind of redo with the modern ways and stuff and whatever. But that doesn't mean it's cheap, because you know, ordering oligos aren't free, and especially if you like in bisulfate, if you're doing pre-methylated oligos and stuff, they're very expensive and so on. You know, it can be a substantial investment to kind of re-synthesize pieces of kits and so on. And you know, commercial things, you know, in the medical side, you have like validated things that have approvals and stuff. I mean, there's all kinds of things that can interact with, like even minor tweaks of protocols are not feasible. But all other things being equal, if you're not, you know, if you have the ability to not fill up a dual, keep it as low density as you can. Avoid short indices. People still run things like TrueSeq 6mers. That's really too short, given the number of sequences you get. The index-based call quality, I think, even kind of went down with the NovaSeq and so on compared to the earlier instruments, which I think were a little more robust against some of these pathologies for low complexity and stuff. And it's just not enough margin, really. However, so like TrueSeq 1 to 48 are 6mers. But for example, if you look at TrueSeq 1 to 48 and empirically look at them as 7mers, if you extend that last base to a 7mer, it actually turns out that there's variation in that 7th base. And that sometimes is critical, because there's actually one of the TrueSeq mers that is like 2 mismatches to phi-x poly G as a 6mer, but it's 3 mismatches apart as a 7mer. And so there's really a gain there to be had from pushing out to the empirically extending those 6s to 7s. Don't be careless with the sample sheet. You know, take care to read your kit's documentation. Make sure you know what instrument you're running on, so you know whether something should be reverse complemented or not, and so on. Just try to get those details right. Yeah, don't try to do anything that basically severely makes the samples in the mix inhomogeneous, right? So that means like no spike ends, use iSeq or something else. You know, don't assume that your sample sheet really reflects reality. I mean, you know, ideally it does, and ideally, even with the blind approach, you kind of get results that look okay, and then you know you kind of think it's okay. If you don't have, you know, the time or resources, or you know, or whatever to go and actually check, then you know, okay, I guess you got to proceed. But you know, be aware that all kinds of things happen, and just be open to various possibilities when you're trying to interpret what's going on when things don't look like they're going well. Do realize that index hopping can occur, misdemux occurs, cross-contamination even outside of the different kind of ways we happen does occur. Evaluate, you know, what kind of downstream analysis you're doing and think about how damaging it would be, what levels would be worrisome, and check to the extent that you can whether those levels are occurring or not, and so on. And yeah, just realize that the blind approach doesn't always work great. You know a lot now about what can happen and so on, and maybe if you do reach a situation where things are happening, you at least can sort of have some hypotheses now that you can chase down to kind of proceed. On the positive side, yeah, pre-flight your mixes. I know that, that can be hard because especially if you're kind of a low fraction of a lane and you're going to be mixing with people that maybe the core is going to help you find or something, then you may not be in direct contact with those people or something. But to the extent you can, I think like some of the cores don't really check so hard. They just mix and you get what you get. Try to the extent possible pre-flight. I do recommend at least looking at your histograms of the indices. If you're going to do one thing that isn't just straight black box blind approach, just look at the tops of those index histograms. They're fairly easy to generate. You can even do those with the FASTQ files that are already demuxed. You got to change the command a little bit, but you can I showed you where the index reads embed into the output file names, and as long as you have all the samples that correspond to the lane and you have the undetermined pile, which they should really give you, then you can actually still get all the index reads that existed in the lane and compose the histogram, even if what you're delivered is purely just a pre-demux set of files. So you can still construct the histograms, even if you're not given a raw run folder. And if you're going to look at one thing, I do recommend looking at those and just see how those look compared to your sample sheet. If you do got dual indices, try to do some kind of full grid analysis because you're going to get visibility into the index hopping and so on. Of course, if you did a dense dual, there's nothing to see there, but try not to do dense duals. Do at least look at the basic indicators, %PF1, %Demux fail, % in your expected, including phi-x. Make sure you always throw in phi-x because it ends up being, if phi-x is 5% of your lane, which wouldn't be that unusual, then and you don't have phi-x as a target in the sample sheet, you're going to see your demux rate go down 5%. And like I said, I normally consider good demux like 98% or 97%. And if you go take out 5% and you're down at 92 or something, to me, that looks like something's missing. And what could be missing is just phi-x or something, right? But at least you should, if you have phi-x as a target, you know how much to ascribe to that piece, right? Whereas if you leave it out, you don't know whether, if you're 10% down and you got like 88%, are you 10% down because phi-x is the 10%? Or are you 10% down because phi-x is 1% and then you've got some mistargeting or something or whatever, and that's the gap. You just don't know until you have phi-x as a target in your sample sheet. Where are we here? We're yeah, use UMIs if you can, but most of us are not really redeveloping protocols, so either the protocol has UMI support or not. If you do have the ability to change protocols to a more modern one that does have UMIs, great. Or just if you're developing a protocol, incorporate UMIs at that time or whatever. But at least you're aware of them and kind of what the goal is and so on. Be very suspicious of all demux piles whose size is small, right? So even if sequencing is perfect and there's no weird base call error, even if base call error was 0 with the index reads, right? Don't forget there's always index hopping. And so that tiny demux pile could exist purely because that tiny demux pile could be highly contaminated with other guys, even just from index hopping. And also from the base call error kind of things, that tiny pile could be purely derived from fuzzes from other guys and not represent any amount of the missing guy, not just a low guy. So, is a low guy low or is he complete missing or a complete failure or something? Well, it's hard to say, right? Sometimes you have enough information in the main reads that you can try to figure that out, but sometimes you don't. But anyway, it's very hard in general to interpret the small size piles. So when you do have the ability to cross-check demux, do that. So if you are amplicons, or maybe some of the samples are from different genomes or something, right? So then just which genome it aligns to kind of at least gives you some information on what sample there it is. When you do have extra information, it is generally advisable to try to cross-check Demux to that. And sort of related to this pre-flight stuff, be careful with who you share a lane, because sharing with somebody whose libraries are crazy does more than just sort of lower your representation by an amount proportional to what they mixed in. They can cause so much damage to the lane that it's hard to recover your stuff out of it at all to any appreciable degree. So the damage can be super linear relative just to the amount that's physically being mixed. So you really don't want to kind of promiscuously mix with someone who's known to sort of like just produce really crazy stuff or something. All right, so last slide. What about kind of ultimate indexing? So this is what I was talking about. So it's basically derived, in part from like this, the fact that the kits aren't really well optimized, and we do have these long indices available now. So maybe ultimate indexing would look something like this, and this would basically take care of almost all the problems we talked about, except for index hopping. But then you do large unique duals, and that mitigates that. So maybe the indexing would look like something. Let's use the bases we got. Maybe we'll do 13 plus 13 for the tags. That's 26 bases for indexing. Of course, we're going to treat that as a unique dual. So we've got basically 13 bases that we're using to separate out guys, and another 13 bases that we're using to defend and detect index hopping. So it's 13 MERS strength for sort of the decision on where to put it. And we can use maybe 5 plus 5 base UMI. That's 10 bases for the UMI. And that's 1,000,000 to 1 extra on top of insert length or whatever else you've got. And remember, we only need to really, the UMI only really needs to help us say you're aligning to the same place on the reference genome. So 1,000,000 to 1 additional randomization is pretty good. And even if the UMIs weren't perfectly randomized, that would degrade gracefully to effectively, instead of 20 bits of entropy, even 10 bits of entropy would still be pretty good there. So you are going to need to have a custom Demux pipeline to take full advantage of these, but whatever. If you're consulting an expert to design these indices, then they can help you with making the pipeline to properly take advantage of them. And then we're going to use the fact that we're probably going to have a very large plexity available, because the 13 MER indices will allow us to have rather distant indices in rather large pools. And we're going to use that to take care of the low plexity problem. And 10x actually is one of the ones that actually seems to appreciate this, because their default is to actually multiply tag every library with 4 distinct indices. And that's the idea, which is you've got so many indices available that you can afford to give multiple of them to each bio sample. And thus, even if you use a single plex or even a 2-plex or whatever, you can kind of guarantee index diversity and make the base caller much happier, because you can essentially force every guy individually to be base balanced. And so no matter what the representation skew ends up being with high ratios or whatever, or somebody does just a 2-plex or something, it just doesn't matter, because every library is individually balanced. Because we just randomly multiply tag him with one of 4 or 16 or whatever tags. And we just, in the Demux piles, we'll just eventually merge those piles into a single sample. But because we have that index diversity and so on, the base caller is much happier, because we're not forcing him into this low complexity world that he really hates. And yeah, so like I said, I think 10x actually learned this lesson, and their routine thing to do is to like A, B, C, D. They have 1 A, B, C, D, 2 A, B, C, D, 3 A, B, C, D. Every sample, which is a single biomaterial, gets 4 distinct tags on it. And this is kind of the reason. Yeah, so maybe if you want a 96-plex, you design a 384 and give each one 4. Then when you have the little tube of adapters that you're going to use to tag the library, that little tube is just a continuous mixture of all those 4 guys. And so each molecule randomly gets one of the 4. All right. And then because we also have a large plexity, we can dedicate a whole bunch of those indices to either control libraries or lane brighteners or whatever you want. So instead of PhiX being this ancient library, which has a dark I1 and this fixed sequence I2 that tends to collide with a lot of other libraries, we just tag it with a whole variety of other things. So we can do we can give it 32 indices or something, right? That are just, and then we just reserve those forever for that thing, right? So it never collides with anybody's user libraries, and it's so diverse, it doesn't matter how much of it you spike in, it's not going to cause low complexity problems or anything. So yeah, one of the great ironies of PHYX is that while it's sort of a main read brightener, it sort of helps the main reads become equal ACGT distribution. It's actually a pathological affier for the index reads because it only has a single index and its indices are rather poor. And so if you, as you spike in more of it, the main reads are getting more and more balanced and the index reads are getting worse and worse balanced. So that's not ideal. And we certainly have seen lanes that have taken damage from amounts of PHYX, and it just doesn't have to be that way. So, you know, like I said, we'll just multi-tag it with some things that we're never going to use for any other libraries, right? So it would never conflict. So anyway, if you do all those things, then basically the only thing we haven't controlled for is index hopping, but we've made a unique dual, and so that'll help. And so actually the bigger you can make the unique dual, the better. So it's better to run like a 96-plex unique dual than it is to run a 10-plex unique dual, because there's just so many more empty squares out there. You know, when you look at a single I1 or I2 hopping, just the rows and columns are bigger. They have more white squares. So the chances that it lands into something that's not you is sort of better. Well, I mean, sort of, because I mean, rows and columns in a unique will only have one spot. So, you know, it's supposed to always kind of miss. But anyway, in general, we do kind of have the feeling that unique duals that are larger kind of do end up being better. You can at least see there's more squares to kind of see at least what's going on there. You kind of get more information just about what the hops are doing. You get a more chance to see any structure in the hops and so on. Anyway, so that's the presentation. I know there's somebody taking it for credit, so you'll get an email tomorrow with a quiz and homework, and then end of next week is fine. Not. I'll send it out tomorrow, and then like it's the Friday after that. Okay, so tomorrow's March 3, and then +7 is 10. So yes. Okay, so I'm gonna stop the recording there.